Welcome everyone. This is one short lecture on the chapter Principles of Inheritance and Variation, which comes in 12th NCERT textbook, chapter number 5. This is very important chapter in terms of NEET examination, and it is a highly complicated chapter, we can tell, because so many concepts come under this chapter only. So with this understanding, we can go with the other chapters like molecular biology, strategies and all. So follow the chapter carefully. So the chapter is Principles of Inheritance and Variation. In the starting of the chapter, into the introduction of the chapter, the author has asked why, if you plant a mango seed, why do you get a mango plant only? And why will elephant give birth to baby elephant only? Why the offsprings, means the children, are similar to their parents? They are not very much similar. They have some similarities with their parents and they have some variations with their parents also. And again, brother sisters also show similarities among themselves, and equally they show dissimilarities also among themselves. So all these questions what we are discussing are, we are talking about a field, a branch of science which is called as genetics. Now in this branch of science called genetics, we'll study about all the explanation for all these questions come from this branch only, which is called as genetics. So what are the answers we want? Why when we plant a mango seed, why do we get a mango plant only? Why elephant will give birth to baby elephant only? Why siblings have similarities among themselves? And why the offsprings have similarities with their parents? Why do they have variations also? All this are dealt under a topic called genetics. Okay, now what is genetics? It's a branch of biology. What do we study under genetics? It's a branch of biology which deals with inheritance and variations. That's what the definition of genetics says. It is a branch of biology which deals with the study of inheritance and variation. Again, there are two new words here, inheritance and variations. Let us try to understand what is inheritance and what is variation. Inheritance means it is a process by which the characters have been passed on from the parents to the next generation. From me to my children, what are the characters which are passed on? That process of passing the characters from parents to the offsprings is called as inheritance. And again, when you talk about variations, what do you mean by variations? Means when I pass on the genes to my next generation, now they don't resemble like me 100%. There are some variations for my children. So that comes under variations. Variations is the degree to which the children or the offsprings differ from their parents. Inheritance is a process by which we pass on the genes to the next generations, and variations is the degree to which the offsprings or the children differ from their parents. Now the study of this inheritance and variation is only called as genetics, right? Then they exploited the variations present in the wild population. Humans knew that the variations are, the reason for variations is sexual reproduction. When do they know this? From 8000 to 1000 BC, humans knew that the reason for variation is sexual reproduction. So that's why they exploited the variations which are seen in the wild populations. So in nature we have so many wild plants and animals. So they exploited the variations which are seen in wild plants and wild animals to get benefited varieties, right? So for example, if you take, they have done artificial selection among the available breed. They have done selection artificially, they have selected a variety. Now artificial selection has been done in carnivorous varieties or domesticated varieties. So they have done artificial selection of domesticated varieties and domestication among the ancestral wild cows they have done, and they produced a well developed breed which is called a Sahiwal cow in Punjab, which is better yielding in terms of milk. Okay, so Sahiwal cow in Punjab is produced by having a knowledge about these variations, and they know that the variations are hidden in sexual reproduction. So by artificial selection and domestication, they have produced a well developed Indian breed of cow which is called a Sahiwal cow, where in Punjab, right? Now humans are doing this, ancestors are doing this, they are developing highly developed high yielding breeds also. So but they don't know the scientific basis. What is that which is responsible for these inheritance? What is that which is responsible for the variations? What is that which is being passed from the parent generations to the offspring generations? So they don't know the scientific basis behind that. That's what the NCERT says. The ancestors knew the inheritance and variations of characters, how the inheritance happens, how does variations happens, they know, but they're not aware of the scientific basis. They're not aware that gene is there which is responsible for this inheritance and variation. That they don't know. Now, who coined the term genetics? A scientist called W. Batsman coined the term genetics. And what is the Greek meaning of genetics? Genetics means to descend. To descend means from me, my children, next generation are descending. So like that. The word genetics was coined by Batsman, and the word genetics in Greek, it means to descend. And then who coined the term gene? What do we study in genetics? We study about genes. Then who coined the term gene? The word gene was introduced by scientist called Johansen. Johansen introduced a word called gene. Who is father of genetics? Gregor John Mendel is father of genetics. But by the time when Mendel was doing experiments on genetics, neither the word genetics was coined nor the word gene was coined. Then what was the word Mendel used for these terminologies? Means Mendel used a word called factor. Now we replace that factor with gene. So Mendel used a word called factor instead of gene. Later Johansen came and he renamed this factor to gene. And then who is father of genetics means who is that person who have contributed significantly in the field of genetics means Mendel. So that's why Mendel is called as father of classical genetics. And who is father of experimental genetics? The father of experimental genetics is Morgan. Mendel is father of classical genetics. Morgan is father of experimental genetics. And who is father of human genetics? The father of human genetics who worked on human disorders, genetic disorders, is Sir Archibald Garrod. Archibald Garrod is father of human genetics. Experimental genetics father is Morgan. Classical genetics father is Mendel. Have a screenshot. We'll talk about certain genetic terminology then. Right. Now, before getting into this branch, genetics, let us see some terminologies which will help us to understand the concepts in a clear manner. Like we have to see what is gene, what is genome, what is phenotype, then what is genotype, what is cross, what is pure line. So these words we need to understand. So we'll have an understanding about that before getting into crosses and all, which were worked out by Mendel. So let us see now genetic terminology. So under that, the first terminology comes as phenotype. Yes. Under that, the first terminology is phenotype. Now, what do you mean by phenotype? If you look at me, so what is my complexion? What is my height? So these are certain phenotypic characters. That means phenotype means it is physical appearance, right? So what is phenotype? It is physical appearance of an organism. And why is my complexion like this? Why is my height like this? It is dependent upon the genes which I have inside, and of course on the environment also. So we can tell the phenotype, which is the physical appearance, is a result of the genes and the environment. So phenotype means the physical appearance is the result of, it is the result of the genes and the environment. Right? And so I think we understood what is phenotype. Now let's go to the second one. What is genotype? What is genotype? It's a genetic makeup, it's a genetic constitution of an organism. Which type of genes I have so that my complexion is like this, my height is like this, my skin color is like this, my hair appearance like this. So what is genetic, what is genotype? It is a genetic constitution of an organism. Genetic makeup you can write, or you can write it is the genetic, excuse me, it is the genetic constitution of an organism. Genotype is a genetic constitution of an organism. And Johansen coined the term gene, right? Let us see what is gene then. The third term is gene. Gene is the structural fundamental physical unit of heredity. What is gene? It is a fundamental, structural and physical unit of heredity. Structural, fundamental, physical unit of heredity. Gene is a unit of heredity. And what is gene? Gene is a unit of heredity, and gene contains a nucleotide sequence which is going to determine for a protein. So what is gene made of? Gene contains DNA. What is DNA made of? DNA is made up of polynucleotides. Nucleotide sequence will be there. This nucleotide sequence, what does it determine? The DNA or the nucleotide sequence will determine the protein. It will determine the protein or the amino acid composition present in a protein. So genes will code for a protein. So particular protein is coded, that's why my complexion is like this. Particular protein is coded, that's why my hair is like this. Particular protein is coded, that's why my height is like this. So phenotype means physical appearance, genotype means genetic makeup, and gene means a structural, functional, physical unit of heredity. And what does the gene has? What is the gene made of? DNA. What is DNA? A nucleotide sequence. What is that nucleotide sequence coding for? A protein. Now let us see next one. What are alleles? What are alleles? Alleles are alternate forms of genes. Alleles are alternate forms of a gene. Alleles are alternate forms of a gene. For example, again, we tell there are two types of alleles. One is dominant allele. The other one is recessive allele. As you are telling, alleles are alternate forms of a gene. What are the alternate forms? One will be the dominant form, the other one will be the recessive form. So dominant form and recessive forms are the alternate forms of the gene. For example, if you take a gene, if you take a character, next one, we'll see what is character. Character. For example, if you take a plant, plant height is a character, flower color is a character, seed shape is a character, seed color is a character. Character means it's a phenotypic character, it's a physical appearance. Character means, for example, if you have to list down, plant characters means height is a character, seed color is a character, flower color is a character, seed shape is a character. Now again we can tell traits. Alternate forms of genes are called alleles and alternate forms of characters are called traits. So what are traits? Traits are alternate forms of character. So again, here we have two traits, dominant trait and recessive trait. Like alleles, we have dominant allele and recessive allele. Here also we have dominant trait and recessive trait. For example, if you take the height as a character, if we take the height as a character, now the height can be very tall or it can be dwarf. So tall and dwarf are traits, right? So for the character called height, what are the traits? What is a dominant trait and what is a recessive trait? Tall is a dominant trait, dwarf is a recessive trait. For the character called seed color, what are the traits? Round shape and wrinkle shaped are the traits of the character called seed color. Seed color, if you take, round is dominant, so this genetic terminology is there in NCERT page number 72 and page number 73. Take a screenshot, we will continue with the remaining terminologies. Yeah. Now what did we discuss so far? We discussed what is genotype, what is phenotype, what is gene, then what are alleles, what is character, what are contrasting traits we discussed, right? So through diagrams, if you have to understand, for example, there is a gene, a gene which is coding for a character. The character is height of a plant. Now, for the character height of a plant, there are two contrasting traits. What are the two contrasting traits means? What are the two contrasting traits for the character height means? Tall and dwarf. Now, tall, what is tall? You are looking at the tall plant, means it is a phenotype, right? Tall represents the phenotype and dwarf also represents the phenotype. And we told the phenotype is because of the genetic makeup. Now means the genes are responsible for the height of the plant, tall and dwarf. Now the gene is having two traits, right? What are the two forms? Alleles. Means the dominant allele is called capital T. The recessive allele is called small t. That means capital T. What is capital T? Capital T is an allele and small t is an other allele. So capital T and small t, what are they? They're alternate forms of a gene. In that, capital T denotes the dominant allele and small t denotes the recessive allele. It denotes the recessive allele. I think we understood now what is meant by a gene, what is meant by a character, what are the traits and what are the alleles. So again, if we have to see what is genome, the complete set of genes which are present in one organism, the DNA content of one complete set of chromosomes is called as genome. We'll tell human genome, right? Excuse me. Human genome. Now, what do you mean by human genome? It is the DNA content present in one complete set of chromosomes. The DNA content which is present in one complete set of chromosomes of an organism, human, or it can be bacteria, or it can be yeast, like that, yeast genome, bacterial genome, human genome, like that. Now again, in this genome, if one set of genes are there, that's called haploid. What is haploid? What is diploid? And what is polyploid? Genome means one complete set of genes, one complete set of chromosomes. Now haploid means it's the same. One haploid means one copy of genes. Haploid means one copy of genes is called haploid condition. Diploid condition means two copies of the genes. Diploid condition means two copies of the genes. So where are genes present? Genes are present on chromosomes. For example, haploid content means only one chromosome. This is the first chromosome, second chromosome, third chromosome, fourth chromosome, fifth chromosome. Only one set of genes are there, then it denotes haploid condition. If two copies of genes are present, that's called diploid condition. Means if you have to show diploid condition, means I have to make them pairs. First chromosome is in pairs, second is also in pairs, third is also in pairs, fourth is also in pairs, and fifth is also in pairs. Now if pairs are there, then that content is called diploid content. Haploid content is represented by N, diploid content is represented by 2N. Polyploidy means many sets, right? One set haploid, two sets diploid, means many sets is called polyploid. By mutations we can generate many sets of genes is called polyploid. Means for first chromosome itself, many are there, many sets is called as polyploid condition. I think we understood what is meant by genome. Have a screenshot, then we will see what is locus. Right? Now locus means it's a location on the chromosome where the gene is located or where the alleles are located. Locus or loci. It is the specific location on chromosome where the gene is located. It is the location on the chromosome where the gene is located. We call it as locus. For example, if we take a pair of chromosomes, first pair of chromosomes, this is diploid condition because a pair is there, a set is there, right? So in this is first chromosome of paternal origin, this is father's chromosome, we call it as paternal chromosome, first chromosome from the father, and this chromosome is maternal chromosome, first chromosome. Now on this pair of chromosomes the genes are located. Take the gene called capital T. Now where it is located, if you tell the location, that's what is called loci. For example, capital T is here, the gene capital T is located there, and here also on the same place in a homologous chromosome, on the same place, either capital T can be there or small t can be there. Either this form can be there or this form can be there. So this is what is locus. So what is locus? It's the specific place on the chromosome where the gene is located. The gene which is coding for the height is called T. So it is located at this particular locus. Now if the genes are located at the same locus, then these two, capital T or capital T small t, they are called alleles. If you take another character which is present here, that is capital R, and here small r, means round and wrinkled. So this is on a different locus, this is on a different locus. Now this gene to this gene, since they are on different loci, they are called non-alleles. Capital T is allele to capital T or small t, whereas capital T is non-allele to capital R or small r. So T and T are alleles, whereas T and R, they are called non-alleles. Understood till here? Now we took a pair of chromosomes, first chromosome, it is in pair. One is from father through the sperm and one is from the mother through the egg. It's called homologous chromosomes. What do you mean by homologous chromosomes? Homologous chromosomes means the chromosomes which are same in structure, composition and behavior. So see, they are same in size, structure and behavior. Homologous chromosomes means it denotes a pair of chromosomes which are similar in size, character and shape. Out of this pair, one chromosome is paternal, father's origin, paternal in origin, and the other one will be from the mother, it will be maternal in origin. So hope you understood what is homologous chromosomes. Okay, so this is one homologous chromosome. If we want to see the second pair of chromosomes, so this is second pair of chromosomes. So these are also homologous chromosomes because this is same like this. But when you compare this chromosome with this chromosome, size is different, shape is different. So then they are called non-homologous chromosomes. This chromosome to this chromosome, they come under non-homologous chromosomes. Now let us see what do you mean by homozygous condition and what do you mean by heterozygous condition. What is homozygous condition and heterozygous condition? In a homologous pair of chromosomes, for example, if capital T is here and on its partner also if capital T is only there, if capital R is here, on its partner also if capital R is there, that denotes homozygous condition. So if both the alleles represents the same, that is called as homozygous condition. If same alleles are present on the homologous chromosomes, that's called homozygous condition. If same alleles are present, where are they present? On homologous condition, on homologous chromosomes. Like capital T is here, again capital T only should be there. Capital R is there, again capital R only should be there. What is the condition called? Homozygous condition. What is heterozygous condition? If capital T is here and on the opposite one if small t is there. If capital R is here, on the opposite one, if small r is there, then that's called heterozygous condition. If different alleles are present on the homologous chromosomes, that's called heterozygous conditions. If different alleles are present, then the condition is called heterozygous condition. Understood? Take a screenshot, then we'll talk about cross also. Mendel conducted crosses in pea plants. Now we should understand what is cross, what is monohybrid cross, like that. We should see that terminology also. We will see, and then we will get into the concept. Now what is cross? So cross is mating between two individuals leading to the fusion of the gametes. So cross, if we have to give the definition of cross, it is mating between two organisms which results in fusion of gametes. Mating between two organisms which results in fusion of gametes is called as cross. Now, if you are doing cross between male and female for one particular character, then that's called monohybrid cross. So you understood the cross. Cross means mating between two organisms, one black and one blue. Mating between two organisms results in fusion of the gametes is called as cross. Again, here we have monohybrid cross. Now, what is monohybrid cross? Crossing between two organisms for one character. Crossing between two organisms with different traits for one character. Crossing between two organisms with different traits for one character. What is the character? Height is the character. For the height, what are the traits? Tall is one trait, dwarf is another trait. So if you take a character called height, for the height, the two different traits are tall and dwarf. If you take a tall plant as male and dwarf plant as female, if you cross them, then such a cross is called as monohybrid cross. Monohybrid cross means crossing between two organisms which are of two different traits. One is tall trait, other one is dwarf trait. So for one character, if you do, it's called monohybrid cross. Next one, dihybrid cross. So we can tell dihybrid cross means crossing between organisms with different traits for one character, or it's for two characters. It will be for two characters. So dihybrid cross means crossing between two organisms with two different traits. With two different traits for two characters. So if you take seed shape as a character and seed color as another character. If you take seed shape as a character, the shape is round and wrinkled. Seed color means yellow and green. So if you are doing cross between the organisms for two different characters, then that is called dihybrid cross. Then there's another thing which is called test cross. Mendel conducted test cross also. What do you mean by test cross? So for example, we told phenotype. What is phenotype? Phenotype is a physical appearance. It is a physical appearance. The physical appearance of the plant is tall. The physical appearance of the plant is tall. Now you want to find out whether it is homozygous tall or heterozygous tall. What do you mean by homozygous condition? Homozygous condition means on a pair of chromosome at a particular locus, it has capital T and capital T only. This is homozygous tall. And what do you mean by heterozygous tall? Heterozygous tall means on a pair of homologous chromosomes, if on one chromosome capital T is there, other chromosome small t will be there. Okay, so if you take a height of a plant, then tall is dominant. So we want to find out the genetic constitution, whether it is homozygous tall or heterozygous tall. Then the cross what we have to conduct is called as test cross. Means if you have to write down what is test cross means, in order to find out the genotype whose phenotype is known. What is the phenotype? Tall. Phenotype is known, but you want to find out the genotype, whether it is homozygous or heterozygous. If you do a cross, that is called as test cross. We will understand this better when we go further ahead. So we have seen here what is cross first of all, what is monohybrid cross, what is dihybrid cross, and what is test cross, right? So then when we are doing these crosses, we represent them in a square which is called as Punnett square. Right, this Punnett square is a graphical representation. Punnett square is a graphical representation of the probabilities of the outcome. When the gametes are fusing, what is the probability of outcome will represent in the form of a checkerboard, which is called as Punnett square. So, which was designed by a British geneticist. His name is Reginald C. Punnett. Reginald C. Punnett, a British geneticist, has designed a checkerboard which is called as Punnett square, which will help us to understand the probabilities when the crossing is happening. This question, Punnett square, was designed by British geneticist. Who designed it? British geneticist. This has been asked in NEET previous examinations. So we discussed the terminologies: what is monohybrid condition, what is dihybrid condition, what is cross, what is phenotype, what is genotype, what are traits, what are alleles, what is character, what is locus, what are non-alleles, all that we discussed. Take a screenshot, then we will talk about Mendel, who is father of genetics, and the contributions made by him. Better understanding all the summary of the genetic terminology I once again took on the board. Let us see this and then we'll go to the next concept. So what is phenotype? Children, phenotype means it's a physical appearance, right? So what is our color, what is our complexion, what is our height, what is our weight, that comes under physical appearance. Now what is genotype? Means why we are like this is because of the genetic constitution, the genes what we have inside. So that's called genetic constitution, is called as genotype. And genes, right? So what are genes? Genes means they code for a protein, they code for a character. Gene is a structural, functional and physical unit of heredity or inheritance. Now genome, the DNA content. Gene has nucleotide sequence. Nucleotide sequence means the genetic material, that's DNA. Now the DNA content present in one complete set of chromosomes is called genome. One complete set of chromosomes. We have 46 chromosomes. 46 are pairs. Now this one complete set means 23 chromosomes. How much DNA do I have in my one complete set of chromosomes? Means how much content of DNA means 3.3 into 10 power 9 base pairs, right? So that is my DNA content per one set of genes. That is human genome. Now if one complete set is there, we call it as a haploid genome. If two sets are there, if paired condition is there, that is diploid genome. 23 chromosomes is called as haploid condition. 46 chromosomes in human is called as diploid condition. We can write haploid condition means 23 chromosomes of human. Diploid condition means pairs, that means 23, it becomes 46. 46 chromosomes of human represents the diploid condition. Then alleles, gene codes for a protein. Now the alternate forms of genes are called alleles. For example, if we take a height which is giving, in that the alleles will be capital T and small t. In that capital T is a dominant allele, small t is a recessive allele. Okay. Now loci. Loci is a place where the genes are located on the chromosome. It is a special location where the alleles are located on the chromosome. Now character. Character is a physical appearance. It is a physical form. If you take the pea plant, in that height is a character, flower color is a character, seed shape is a character, seed color is a character. So they are called characters. Now all the plants will not be of same size. Some plants will be very tall, some plants will be dwarf. So tall and dwarf are different forms of a character. They're alternate forms of a character. So then we call them as contrasting traits. The character, if you take height as a character, what are the contrasting traits? Tall and dwarf. Like that we took gene. Alternate forms of genes are called alleles. Dominant allele, recessive allele. In the same manner, character, height. The height, what are the contrasting traits? Tall is one trait, dwarf is one trait. Now non-alleles. Alleles means which are present on the same loci on the same chromosome. Non-alleles means if a gene is present on the top of the chromosome, another gene is present on the bottom of the chromosome. So they are at different loci. Then they are called non-alleles to each other. homologous chromosomes. Homologous chromosome means a pair of chromosomes. If I take these two things, so this is a pair of chromosomes which are similar in size, structure and behavior, then we call them as homologous chromosomes. Now if I take another pair like this, now this is different from this in its size, structure, color, complexion, everything, so then they are called heterologous chromosomes. Homologous chromosomes means which are same in structure, size and behavior. Heterologous chromosomes means which are different from this one is called as heterologous chromosomes. Now in that homozygous condition, on this homologous chromosomes, now the genes capital T are there, both capitals are there, capital are there. So if the same alleles are present on the homologous chromosomes, that's called homozygous condition. If different alleles are present, means capital T and small t, capital R and small r present, that's called heterozygous condition. Then cross, Mendel conducted cross in pea plants, right, to check the probabilities. Now cross, what is cross? Cross is mating between two individuals. When the two individuals mate, then the gametes will fuse, then the offsprings are formed, siblings are formed. That is called as cross. Now when you do cross between organisms for one character, then that is called monohybrid cross. If you do cross between organisms for two characters, then that's called dihybrid cross. Now when we do the cross, we get offsprings. To check the probability of the offsprings, so we does Reginald C. Punnett, a British geneticist, has designed a Punnett square which will help us to check the probabilities. So all this is genetic terminology. Now we will see what are the contributions Mendel made towards the field of genetics. Take a screenshot. Now let us discuss about Mendel. So without discussing Mendel, studying genetics is not appropriate. So why? Because Mendel is called as father of genetics. He has contributed significantly in the field of genetics. Now his complete name is Gregor John Mendel. So he was born in 1822 and he passed away in 1884. Now out of his poor academic background, he joined Augustine monastery as a monk, as a priest. He joined Augustine monastery at St. Thomas, which is in Brunn, as a monk. Now it is in Czechoslovakia. Now later from that monastery, he was sent to University of Vienna to study natural science and maths. So the people have sent him to study natural science and maths. He studied natural science. After doing his graduation in natural science and maths, he returned back and he joined as a sub teacher in the same church. Now Mendel, he observed pea plants in his church. He observed two different varieties of pea seeds and he developed interest in studying genetics and the patterns of inheritance, and he selected pea plant only as his reference material to study genetics. Now Mendel initial works, he thought of working on 34 characters of pea plant, but then he felt like 34 characters are too many characters, then he confined to 22 characters, but finally he worked only on 7 pairs of characters. He worked only on 7 characters: height, shape, flower color, seed color, pod shape, all those things. How many characters did Mendel work? He worked on 7 characters on the pea plant. How many years he worked on these 7 characters? Means 7 years he worked on these characters. What are the years? From 1856 to 1863, 7 years. This question has been asked in NEET examination. Mendel's experimental material is pea plant. Mendel studied how many characters? 7 characters. Each character has contrasting traits, means how many varieties are there? 7 twos, 14 varieties are there. And Mendel conducted experiments on pea plant for 7 characters or 14 traits for 7 years. From which year to which year? It is from 1856 to 1863, 7 years. And whatever experiments he did, he submitted his observations, his ideas in a local journal. The name of the journal is Proceedings of Natural History, Society of Brunn. In the 4th volume of the journal, Proceedings of Natural Society, History of Brunn, there with a title called Experiments on Plant Hybridization. The title of experiment what he has kept is experiments on plant hybridization. He published whatever results he got from the experiments what he has done, monohybrid cross and dihybrid cross experiments what he has done. But Mendel's works remain unrecognized. When did he publish it? In 1865 he published it. In 1863 he completed. In 1865 he published it. But his works were remain unrecognized. What are the reasons for the Mendel's works to not to get recognized? So if you have to list down the reasons why his works are not recognized, means first of all, he's not a scientist. He's not a scientist because he's a priest, he's a monk, so the scientist community so feel difficult to accept him as a scientist, and his attitude or his nature is not aggressive so that he can convince the people. So that's the second reason. And Mendel published his whatever his reference are, his conclusions in a local journal, and at that time print media is not vigorous. So since it's a local journal, it has been distributed in local areas only, it did not go outside. Now next one, Mendel is a person, because he has studied math and he has studied natural science, he is the first biologist we can tell who applied mathematical logic and statistics to solve the biological problems, whereas the other biologists who don't have this maths background, so understanding maths is completely new for them, so that's why they couldn't step inside to see what Mendel has done. Okay, then next, Mendel's genes. Mendel considered gene or factor as a stable discrete unit of inheritance, and he tells that these genes or the factors are being stably passed on from one generation to other generation. But by the time people were too much influenced by Charles Darwin's theory of natural selection, where he talks about variations. Natural selection, variations will be there. When people are too much convinced with variations, they were unable to accept this thing what Mendel is telling, so they are constant, they're stable, and stably passed on from one generation to other generation. And so science needs proof. But because Mendel is working on factors and genes, if the scientists have asked, show us the proof, show us the alleles, show us the genes and chromosomes, so you can see that only in under microscope. So at the time, since microscope is not designed or discovered, he was not able to show the physical proof. For these reasons, Mendel works are remain unrecognized. What are the reasons? Print media was not popular at that time, and he published his article in a local journal only, and he is not scientist, so that's why people did not accept him. His personality is not aggressive to convince them. And the other one, Mendel is the first person, being a biologist, he applied mathematical logic to solve biological phenomenon. Biologists did not understand that. Next one, Mendel told that genes or factors are stable and discrete units. Again, people were at the time were convinced by Hugo de Vries' theories of variations, and Mendel finally he couldn't give any physical proof. For that reason also, like science will not accept anything without a proof. So because of all these reasons, Mendel works are not recognized, and he died in the year 1884 due to kidney failure. He died in the year 1884 due to kidney failure. After 16 years of his death, means 1884 plus 16 if you take 1900. In the year 1900, 3 scientists independently from 3 different countries did the same work and published the same work. What are the names of those scientists? In the year 1900, 3 scientists independently rediscovered the Mendel's works, or told the same thing what Mendel already told. So first scientist is Hugo de Vries from Holland. The second one is Karl Correns, we can tell Karl Correns from Germany. And third one is Paul Ehrlich from Austria. Paul Ehrlich from Austria, Karl Correns from Germany, and Hugo de Vries from Holland. These 3 people, they worked on the plants and they gave the same conclusions what Mendel gave. So when they started publishing it, the scientist community told like these works are initially done by some other scientists. Now these people, that means they rediscovered the Mendel's works again. What Mendel told, 3 scientists independently told the same things again. So then they told it's not our contribution, it's a Mendel who has contributed to the field of genetics, and then Mendel was made the father of genetics. Take a screenshot. We'll continue with the 7 characters what Mendel has worked, and how did the pea plant helped him to reach his success. The 7 characters what Mendel finally selected and worked experiments. I have already taken the 7 characters on the board. Let us see. Now, pea plant has 7 pairs of chromosomes. Pea plant has 7 pairs of chromosomes and Mendel studied 7 characters. Now we need to see those 7 characters are present in which chromosomal numbers also. So here I wrote the chromosomal numbers, means this particular character is present on chromosome number 4, this character on chromosome number 1, these 2 characters on chromosome number 4, this character on chromosome number 5, first chromosome and 7th chromosome. So how to remember the order? So we will make a small thing, like if we look at a plant, what anyone looks into the plant first is the height of the plant. That means take height as a first character. Now afterwards, what do we see? The flower, right? So that means take flower color as a second character. After looking into the flower, we will see where is the flower present, at the tip or at the axis, means flower position, take it as a third character. Then flower gets converted into fruit. The fruit of pea is called legume or pod, means pod color pod shape is a 4th character and pod color will be the 5th character, and pod contains seed, so seed color is the 6th character and seed shape is the 7th character. Like that you remember the order. See in which order we have to remember. When we look at the plant, what do we see first is the height. Stem height is the first character. After height, what do we see next is the flower. Flower color we see. Flower color will be the 2nd character and flower position we see, that's the 3rd character. Flower gets converted into fruit. Then fruit of legume, fruit of pea is pod. So we'll see pod shape. So pod shape is a 4th character. Pod color is a 5th character. Pod or the fruit contains seed. So seed color is a 6th character and seed shape is a 7th character. And where are they present? So remember the chromosomes like 4144517. Like a mobile number, you remember, it's like 4144517. That means the first character, stem height, is on the 4th chromosome. The second character, flower position, flower color, is on the 1st chromosome, and flower position and pod shape are on the 4th chromosome only. The 5th one is the pod color, is on the 5th chromosome, and seed color is on the 1st chromosome. The 7th character, seed shape, is on the 7th chromosome. So 5th one is on the 5th chromosome itself, 7th point is on the 7th chromosome itself. Remember, like 4144517. And these are the contrasting traits of a character. For the height of a plant, the trait is tall plant and dwarf plant. For the flower color, the traits are violet is dominant and white is recessive. And for flower position, axial position is dominant and terminal position is recessive. Now pod shape, full, if the pod is full, it is dominant. Tall plant means we have to take it such a tall plant. Dwarf plant means small plant. Dwarf plant is recessive, tall is dominant. And flower color, if we see, violet color is dominant. So if the flower is violet color, it is dominant, and if the flower is white, if it is white in color, it is recessive. Then flower position, if the flower is at the axis of the leaf, if the flower is at the axis of the leaf, that is axial, that is dominant. And if the flower is at the terminal, at the tip, that is recessive. And pod shape, if you go, if the pod shape is full, this is a full pod. If the pod shape is full, then that is dominant. And constricted pod means if the pod is like this. If the pod is constricted, this is recessive and this is dominant trait. Then next one, pod color, green pod. If we buy French beans or flat beans or broad beans or cluster beans, we buy them in green color, means green is a dominant color. So in pod color, green is dominant and yellow is recessive. Coming to seed color, when we see the seeds, yellow seeds, right? So if we take dals, moong dal, arhar dal, all these dals will be yellow only. Means seed, if you go, yellow is dominant and green is recessive. Pod, if you go, green is dominant and yellow is recessive. Then seed shape, when we see the shapes of the seeds, usually round we find, means round seed is dominant and wrinkle seed is recessive, and yellow is dominant, green is recessive for seed color. These are the 7 characters what Mendel has studied in the pea plant. And Mendel conducted all his hybridization experiments in pea plant. So what are the advantages that the pea plant gave for Mendel? Means pea plant is an annual plant, means it will complete its life cycle in a short span, so we can can observe the readings very fast. It is easy to cultivate, doesn't require too much care. That's the first advantage. Second, it's an annual plant. The readings will be very fast. And the third one, pea plant can be both self-pollinated and cross-pollinated. It's a bisexual flower. If you want to do self-pollination, so androecium, gynoecium are nearby, it can undergo self-pollination. If you want to do cross-pollination, then we have to emasculate the anthers, bring the pollen grains and drop on it. So it can be easily self-pollinated and cross-pollinated, so that your required varieties can come. That's the third advantage. And when you're doing hybridization, the F1 hybrids are fertile. So that means when you soak the seeds, when you get the next generation plants, all of them are fertile. This is the major advantage. And the pea plant has number of contrasting traits. Initially Mendel selected 34, then thought of 22, then finally fixed to 7 pairs of characters. So means there are so many contrasting characters in pea, so which is again an advantage. And pea plant contains number of detectable contrasting characters we discussed. Pea plant produces large number of flowers, and those flowers will get converted into seeds. That means his sampling sizes can be huge and enormous, because each plant produces more number of flowers and seeds. So these are the advantages for selecting pea plant, and these are the 7 characters what Mendel has studied. So they can ask you, this particular character is on which chromosome, this particular character is on which chromosome. For this particular character, what is the dominant trait? For stem height, tall is a dominant trait, dwarf is a recessive trait. For flower color, violet is a dominant trait, white is a recessive trait. So for flower position, axial means at the axis of the leaf is a dominant trait, terminal at the tip of the plant, if the plant is having flower, it is a recessive trait. So this also they can ask you, or they can give a question, identify the odd man out, identify the mismatch. In that I will give 3 options which are dominant, 4th option will be recessive. You need to identify which 3 are dominant. So this is not dominant recessive, so this is the answer in that manner. So for all the 7 characters, children, you should understand, you should remember what are dominant traits and what are recessive traits. Take a screenshot, then we will discuss further. Before Mendel, many other scientists also conducted hybridization experiments, not in pea plant but in other plants. Then why couldn't they propose the laws of genetics? Why? What made Mendel to propose the law of genetics means all other scientists they aim to work many characters at a time. Since they were working on many characters, they were not able to give conclusions. But whereas Mendel, if you see, he worked only 1 character or only few characters, 2 characters at a time, so that he can get the accurate data. So if we have to see what made Mendel success in his experiments, so these are the points. First success is selecting pea plant, because pea plant has so many contrasting traits which are easily seen with the naked eye. That is one advantage that Mendel has done. Second, he restricted his experiments to 1 or few pairs of characters only. So he did only monohybrid cross and dihybrid cross. He did not see all 7 characters at a time in one experiment. So he restricted his data to only 1 or few characters at a time. That's the second advantage. He kept track of each character separately. So means monohybrid cross, whatever ratio he got, he kept the track record of that separately. When he connected monohybrid cross for second character, that record, that data, he kept it separately. And since he has mathematical background, he started his experiment with large sampling sizes, 3000, 5000 true breeding plant varieties he will take. And his sampling sizes, F1, F2 generation also will be more in number only. And if we have to count them, it will not be possible. So that's why he used mathematical data, statistics, all those things which helped him to maintain accurate data. Since he had mathematical background, he kept the track record of each and every character quantitative data separately. He verified his results by further experiments. He did not stop his experiments with F1 and F2 generations. He verified the results with F3 and test cross experiments also. So all these things helped Mendel to come out with the conclusions to lay the laws of genetics. What are the reasons which made Mendel to success? The first point is selecting pea plant, of course. The second one is he restricted his experiments to 1 character at an experiment. Others did so many characters at a time. The third one, he kept the track, he kept the data of each experiment separately. Since he has mathematical background, so his sampling sizes were large and he used mathematical logic and statistical logic to solve the ratios. Then he verified his results further with not only F1, F2, with F3 and test cross also. All this helped him to lay the laws of genetics. First law of genetics is called law of dominance. Second law of genetics is called law of segregation, and the second set of generalizations called law of independent assortment. That's what Mendel has proposed. Now, Mendel selected how many pairs? 7 pairs of characters. So, 7 pairs means tall and dwarf is one pair. Violet and white is another pair. Round and wrinkled is another pair. Yellow and green is another pair. Full and constricted is another pair. Means how many varieties you will get? 14. Mendel selected 14 true breeding pea plant varieties as pairs. So, 14 true breeding pea plants as pairs is selected, which are similar for 6 characters. The 7th character will be different. Means which are similar, all of them are having dominant for 6 characters. For 7th character, which is under observation, one plant will be tall, the other plant will be dwarf. So like that he has selected. And here came true breeding pure lines. We should understand what is true breeding. What are pure lines? Mendel's parents are pure lines. How to obtain pure lines? How to obtain true breeding lines means, if you take a plant and if you continuously do self-pollination for it, then it has the same set of genes and there are no genes which are not appearing. Only those genes which are present there will be appeared. So such type of plants are called as true breeding varieties. They are called as pure lines. The plants which have undergone continuous self-pollination for the past 4 to 6 generations. The plants which have undergone continuous self-pollination for the past 4 to 6 generations are called as pure lines. Whatever the genetic constitution is there, it will be the same. There are no hidden genes in it. So Mendel selected pure lines as the parents. So whatever the genotype and phenotype, only that is there. So with that he started his experiments. Take a screenshot, then we will talk about inheritance of one gene. We'll talk about monohybrid cross then. Mendel conducted monohybrid cross. Monohybrid cross can also be called as inheritance of one gene. Means, what is monohybrid cross? Mendel first conducted monohybrid cross, then later he conducted dihybrid cross. If we have to give what's the definition of monohybrid cross means, it is a cross between two organisms, one male and another female, with different traits for one character only. For example, let us take height of the plant as a character. What is that character? Character is height of pea plant. If you take height of pea plant, then Mendel need parental varieties. Now he needs a male and he needs a female. So one parent is a tall plant. One parent, what he has selected is a tall plant, and another parent, what he has selected is a dwarf plant. This is tall in appearance, this is dwarf in appearance. This is tall phenotypically and this is dwarf phenotypically. And we told Mendel parents are true lines. They are pure lines. So they are produced by true breeding. What is meant by true breeding? Which have continuously undergone self-pollination for the past 4 to 6 generations. So that means, so tall and dwarf are the phenotypes. If we have to look at the genotypes, the genotype of tall, since it is pure line, it is homozygous then. Homozygous means it will be capital T only. And dwarf is also pure line, means it is also homozygous. That means the dwarf plant genotype will be small t. So these are the parents what Mendel has selected. He selected the male plant as capital T tall. He selected the female plant as small t dwarf. Now when they produce gametes, now capital T will produce a gamete. Since it is male, it produces pollen grains. Now the pollen grains are of capital T, gametes, right? So the male gametes are capital T, and the female gametes, if you see, female gametes means they are the eggs. So then the female gametes, since they are small t, they'll be small t, and small t gametes are haploid. Now, if we have to see the combination, if the pollen grain comes and fuses, so 2 gametes on the top, 2 gametes on the left side if you write, then the Punnett square has a possibility of 4. Now the possibility will be capital T, small t, capital T, small t, capital T, small t, capital T, small t. Now this generation is called as F1 generation. Here is a parental generation, and fusing, crossing between the parents. Crossing between the parents means you are collecting the pollens from the tall plant and dropping them on the dwarf plant, means we are doing cross pollination. That's what I wrote cross symbol here. Crossing between the parents will give F1 generation. Now capital T, homozygous tall will give 2 gametes called capital T, small t, dwarf plant will give 2 gametes called small t. So the F1 generation, it gives hybrids. So in F1 generation, all F1 plants are hybrids. Why am I calling them as hybrids? Because capital T is also there, small t is also there, so we are calling them as hybrids. Then if we have to give the phenotypic ratio, what will be the phenotypic ratio and what will be the genotypic ratio? So phenotypic ratio means physical appearance, right? It's a physical appearance. Since capital T is dominant, if capital T dominant allele is there, it'll express its trait only, means tall only will be there. That means because capital T is there, the plant is tall. Since capital T is there, the plant is tall. Here also tall, here also tall. Means in F1 generation, all the hybrids are tall. All tall. The genotypic ratio, all of them are capital T, small t's only. So all of them are heterozygous tall. All of them are heterozygous tall. So he got F1 generation like this. These F1 generation plants are fertile. So he got the seeds of F1 plants, and these F1 seeds, when they are grown, he got F2 generation. Now how to write F2 generation? Means if you take the F1 plant, the F1 plant is like this. If the F1 plant is self-pollinated, if the F1 plant is selfed, then you will get F2 generation. For cross-pollination, what we have done? For cross-pollination, if you have selected the male plant, this is the flower. From the male plant, this is a gynoecium. These are the stamens. From the male plant, we will collect these pollen grains. From the male plant, we will collect the pollen grains, and if it is the female plant, from female plant, what should we do for the female plant? These pollens only should go and pollinate. Means in the female plant, only gynoecium is kept intact. The androeciums are trimmed off. Here is the androecium. So you have to bring a scissor and you have to cut the anthers. Removing the anthers from the female plant are must. So this process of removing the anthers is called as emasculation. It's called emasculation. After emasculation, in order to prevent the unwanted pollens to come and drop on the stigma, we have to bag it. Emasculation followed by bagging. Afterwards, we'll bring these pollens from the male plant and then we'll drop it and then we will rebag it. So that cross-pollination only will happen. After doing that, we got all the F1 hybrids they are hybrids, they are heterozygous tall. Now F1 plants, when we self, what do we get? We'll see that. So parental generation, they are true lines, they are pure lines. Means one parent is capital T, another parent is small t. This has produced a gamete like this, this has produced a gamete like this. On cross pollination, we got F1 generation. All F1 generations are capital T, small t. They means F1 generation, they are hybrids. All tall they are, and all are heterozygous. All are heterozygous. In one, capital is there and small is there, we call all of them are heterozygous. And F1 generation are selfed. If F1 generation are selfed, then what do we get? So male gametes will it will form capital T and small t. Since again it is the same plant, in female also we get the same possibility, capital T and small t. Now since they are selfed, capital T small t only combination, so male also will give one capital T gamete and one small t gamete, female also will give one capital T gamete and one small t gamete. So with 4 gametes the possibility of Punnett square will be 4. Now if we see, if we had to fill the genotypes here, capital T will come here, capital T small t will come here, capital T small t will come here, small t will come here. So this is the Punnett square. Now this is called as F2 generation. In F2 generation, if we have to write down the phenotypic ratio, how does it appear? And if we have to mention the genotypic ratio also, how does it appear? So we know that if capital T is there, then the plant will be tall only, means capital T is tall. Here also, since capital T is there, the plant will be tall only. Here also, since capital T is there, the plant will be tall only. Because of small t, it will be dwarf. It will be dwarf. Means if you check the phenotypic appearance, how many tall and how many dwarf? 1, 2, 3 tall and 1 dwarf. Means the phenotypic ratio will be 3 tall and 1 dwarf. And the genotypic ratio, we will see what is the genotypic genetic makeup. One is capital T. This and this are capital T small t's, and one will be small t. Means the genotypic ratio is 1:2:1. So the genotypic ratio is 1 capital T, 2 capital T small t and 1 small t, or simply we can tell 1:2:1 is a genotypic ratio, 3:1 is a phenotypic ratio. So what Mendel saw after doing monohybrid cross, means after doing F1 and F2, he saw that the dominant one is expressing more. The dominant one is expressing more, 3 times it's expressing, and in F1 generation only dominant trait has appeared. The recessive plant did not come. All are tall only, we told. That means the recessive one did not come. So this plant is a tall plant. Capital T is tall, and small t is a dwarf plant. So all the F1 generation plants resembled one parent only. All the F1 generation plants are tall. They exactly resemble this tall parent. They did not turn to be dwarf. And when F1 plants are selfed, he saw 3 times tall plants came and in 1 proportion dwarf plants came. So in F1 generation dwarf plants are not there, but dwarf plants appeared in the F2 generation, and the tall and dwarf plants in F2 generation resemble the same height of the parents only. They are of same height of the tall plant. The dwarf is also the same height of the dwarf plant. So that's what he tells, like the genes had passed on from parent generation to F1 generation stably as discrete units. They don't blend with each other. So there's no intermediate size. Same height tall will come, same height dwarf will come. Intermediate sizes are not there. That's what Mendel has observed. So take a screenshot, then we will talk about the laws what Mendel has proposed related to monohybrid cross. Now if we have to discuss the key points of monohybrid cross, so Mendel found that the phenotype in F1 hybrid, the phenotype of F1 hybrid is tall, the genotype is hybrid, capital T small t, but the phenotype is tall only, and that tall plant resemble the parent tall. Mendel found that the phenotype of F1 hybrid, capital T small t, is exactly same as the parental tall variety, which is capital T in appearance. Then he proposed that in a pair of dissimilar factors, because F1 is hybrid, dissimilar, capital T is there, small is there. In a pair of dissimilar factors, one factor or one allele dominates the other one. Mendel used a word called factor instead of gene. He told that in a dissimilar pair of factors, means in this, one dominates, the one which is dominating, which is appearing, is called dominant form, and the other one is called as recessive form. Now for our convenience, we are symbolizing them with English alphabets. For dominant form, we are taking the capital letter, and the same if you are taking for dominant form tall, you are taking capital T. For recessive form, we'll take small t. We'll not go dwarf D. So you have to fix with one alphabet, and for dominant form, we'll write capital letter, and for recessive form, we'll write small letter. Means for tall and dwarf, we are writing capital T, small t. For flower color, violet is dominant, right? White is recessive. So we will write W. We'll write W and capital W and small w like that. For our convenience, we are using capital case and lowercase alphabets to represent the dominant alleles and recessive alleles. F1 plants were selfed. Parents are crossed to produce F1 plants, whereas F1 plants are selfed. When the F1 plants are selfed, then the gametes are produced in equal proportions. The gametes are produced in equal proportions. 50% of the gametes will be capital T and 50% of the gametes will be small t. 50 percentages, half capital T and half small t will come. When we are fertilizing the gametes, capital T pollen grain has 50% chances to pollinate capital T egg, and small t pollen grain will have 50% chances to pollinate small t egg. That's what NCERT says. Now, as a result of random fertilization, if you see the F2 Punnett square, in F2 Punnett square, so the zygotes formed are one capital T, two capital T small t's and one small t. So as a result of random fertilization, the resultant zygotes appear as the genotypes having capital T 1/4 and 2 are capital T small t's, means 1 second is capital T small t and 1/4 will be small t. So how did you came to know this? By drawing Punnett square. So from the Punnett square, it is evident that random fertilization leads to 1/4 capital T and half capital T small t and 1/4 small t. Out of 4, 1 is capital T, means 1/4 is capital T, 2 are capital T small t's. So out of 4, 2 means half, right? So 1/2 is capital T small t. Out of 4, 1 is small t. So 1/4 will be small t. So this, if you have to condense in mathematical form, then the equation goes like ax plus by whole square. The mathematical condensable form, it goes like ax plus by whole square has the gametes bearing gametes capital T and small t in equal proportion. Capital T will go 50 times, small t will go 50 times. Now, if we have to represent these values in that form, then we have to write it this way. Ax plus by whole square, we will write it as 1/2 capital T plus 1/2 small t square. Right? So now if we have to write ax plus by whole square, if we have to represent it, so we'll represent it as 1/2 t plus 1/2 t whole square, means we can represent it 2 times, right? So when we are representing twice, so instead of square, I wrote it 2 times. Now when we have to expand it, so 1/2 into 1/2 tt means you will get 1/4 capital T, right? Plus, so it is like a square plus b square plus 2ab. A square means it is capital T. B square means small t. 2ab means 2 capital T and small t, right? So a square means it is 1/2 capital T, you will get 1/4 capital T. And b square, if you write, it will be 1/4 small t. 2ab you have to write. So 2, what is a? A is 1/2 t, right? So 1/2 capital T into b means 1/2 small t. This and this will cancel, then you will get 1/2, right? So the simplified form is 1/4 capital T plus 1/4 small t plus 1 by 2 capital T small t. That's what we got in Punnett square proportion also. 1/4 is this random fertilization led to the formation of 1/4 capital T, 1/4 small t and 1/2 capital T small t. Mathematically also it is coming the same thing when you represent in the form of ax plus by whole square. So A represents this one, B represents this one, whole square means 2 times here, right? So I opened it and I kept it. So when you have to multiply it, you will get the same thing again. So take a screenshot, then we'll talk about the laws which talk about the Mendel's monohybrid cross. The next topic is incomplete dominance. Incomplete dominance and codominance, they are not Mendelian inheritance. They come under a separate topic which is called as gene interactions. What do you mean by gene interactions? Means the modification of the phenotypic expression of a gene by the other gene. Means the other gene has impact on this gene. Then we call they are interactions among the genes. For these gene interactions only we can take 2 examples. One is incomplete dominance and the other one is codominance. Mendel talked about dominance, right? Law of dominance, which is first law of genetics. So he did not talk about incomplete dominance. That's why I'm telling this is deviation from the Mendelian genetics. So in incomplete dominance, what happens? What is the example for incomplete dominance? Snapdragon, it's also called dog flower. Its scientific name is Antirrhinum majus, is one example. And 4 o'clock plant, which is called Mirabilis jalapa, is another example for incomplete dominance. And the concept of incomplete dominance was first discovered by a scientist called Correns. Now in incomplete dominance, it is a condition in which the alleles of a heterozygote interact among themselves. Capital R and small r will interact and they will change the phenotypic expression and change the expression. Means we can say here that the allele is incompletely dominant. It's not completely dominant. What do you mean by dominant allele? The one which can express even in the recessive form, the one which can express even in the heterozygous condition is also called as law of dominance. But here it is not expressing completely, it's expressing incompletely. So when we look into the example, I think you'll get better understanding. Now coming to the second example for gene interaction, it is codominance. In codominance, both the alleles express independently. Means both the dominant alleles will be there and both of them they express independently and they give rise to a new phenotype. It is called as codominance. Example for codominance is ABO blood grouping and MN blood grouping. Example for incomplete dominance is Snapdragon plant, which is called Antirrhinum majus, 4 o'clock plant, which is called Mirabilis jalapa. Take a screenshot. We will look into incomplete dominance in detail. Right. So when we see this plant, which is an example of incomplete dominance, there are 2 varieties of the flowers. Yeah, so we are going to talk about incomplete dominance in detail. Now in incomplete dominance, when we have to take the parent plants which should be pure lines, so we have to take a flower which is red in color. So red flower has having the alleles capital R, and another parent is having white color flowers. The white color flowers is having recessive genes small r and small r. So if we have to take the plant Antirrhinum majus or Mirabilis jalapa, the parents are pure lines. One parent will give red color flowers, another parent will give white color flowers. So when we have to cross them, when you have to make the gametes, this will give a gamete which is of R type and this will give a gamete which is of R type. When we do crossing between these 2 plants, then the F1s will be hybrids. The F1 will be hybrid, means one this capital R and small r will come. And so when we talked about Mendelian inheritance, there we told the F1 hybrid resembles one parent. They resemble the tall parent, they resemble the dominant parent we told. But here this F1 hybrids, they neither resemble this parent nor this parent. Means the F1 hybrids are not going to be of red color nor they are going to be of white color. In fact, they are of pink colored. The F1 hybrids are pink colored. So that's what we told, that the capital R is incompletely dominant over small r. Means capital R in the presence of small r is not able to be completely dominant. So that's why it's not giving complete red color. It's giving a mixed color which is called as pink color. Now if F1 hybrids are selfed, if F1 hybrids are selfed, then we'll get F2. It will give two types of gametes. One is capital R and the other one is small r. Again, when we see the two types of gametes, capital R and small r. So these are for male gametes and these are for female gametes. Now the Punnett square goes like combination. We are going to get 4. Capital R with capital R will be one combination. Capital R with small r is another combination. Capital R with small r is another combination. Small r with small r will be the other combination. So this is F2 generation. Now in F2 generation, we got capital R, capital R. As we have seen here, capital R denotes reds. Here also it will be red color flowers. We already saw that small r means they will be white in color. We got small r here, it will be white in color. And these two are hybrids, which is one capital R and small r, one capital R and small r, it will be pink. So we got two pink color flowers in F2 generation. Now if we have to mention the phenotypic ratios, if we have to mention the phenotypic ratio for F2, so if you can see there, 1 is red, 2 are pink, and 1 is white. Means 1 is to 2 is to 1. And if you have to tell the genotypic ratio, 1 is capital R, 2 are capital R, small r, 1 is small r. So one thing to notice here, that in incomplete dominance, the first point is the F1 hybrids will not resemble both the parents. It's not resembling this parent also, it's not resembling that parent also. One difference. And the second difference is the F2 ratios are deviating from the Mendel monohybrid cross. In Mendel monohybrid cross, the phenotypic ratio is 3:1. But here the phenotypic ratio is 1:2, 2:1. And the phenotypic ratio and the genotypic ratio are the same, 1:2, 2:1 and 1:2:1. So this is a concept of incomplete dominance. The examples are Antirrhinum majus and Mirabilis chalapa. Take a screenshot, then we will talk about what is dominance. Explanation for the concept of dominance. So Mendel told that there are certain alleles which are dominant alleles and there are certain alleles which are recessive alleles. Now let's try to understand and give the explanation in a better manner for the concept of dominance. So this is the NCERT information, I took it on the board. Now what exactly is dominance? We are going to try to understand what exactly is dominance and why some alleles are dominant over the other allele. If you have to understand this, first you should understand what a gene is. Gene is a sequence which will code for a particular protein. That's what the NCERT says. Every gene contains information to express a character or a trait. Every gene contains information to express a trait. And in diploid organisms, there are 2 copies of the genes. Since a pair of chromosomes are there, there are 2 copies of the genes. That means there are a pair of alleles. Now it's not always compulsory that the pair of alleles should be homozygous dominant or homozygous recessive. There are cases where the alleles can be one capital and one small, means one dominant and one recessive. Such condition is called heterozygous condition. Now in a pair of alleles, it's not compulsory that it should be dominant only, it should be recessive only. Heterozygous condition can also be there. Heterozygous condition means one dominant and one recessive. So now both the alleles need not be identical. As in heterozygous condition, if you see, one will be capital T, other one will be small t. Now one of that is due to the changes that it has undergone due to mutations. Now why is this small t? Why it's not capital T means one allele is capital form, the other one has undergone mutations, the other one has undergone changes due to some mutations. Because of that, this allele got modified. This is an unmodified allele, and this allele, because of mutations and because of some changes, it has undergone mutations and it becomes a mutated allele. It becomes a modified allele. Since it is a modified allele, the information which is present on this allele is changed. So which is modified? The small t is a modified allele. So the information present on this particular modified allele is changed. To understand, let us take the gene which is coding for an enzyme. A gene is there which is coding for an enzyme, right? So example, gene which codes for an enzyme. A gene has two allelic forms. One is a capital form, the other one is a small form. Now the one which is the capital form is unmodified allele. The one which is the capital form is the unmodified allele. It is a normal allele. It will code for the normal enzyme. Normal enzyme will help in transformation of substrate to the product, and you can see the phenotypic expression. The one which is normal, which is unmodified, produces a normal enzyme. It will make normal enzyme as usual, which is needed for transformation of the substrate into product. Now the other allele which has undergone modifications, which has undergone mutations. So theoretically, if you see, what is this modified allele? The modified allele, what is the fate of the modified allele? The fate of the modified allele can be any one among three. The first one, the mutation, what happened? It can be a silent mutation, means mutation happened but you cannot see the change. So in such cases, what happens? The modified allele also will produce the normal enzyme only, or little less efficient enzyme. That is one possibility. So it's going to produce normal enzyme or less efficient enzyme. That is one possibility. Second possibility is it will do transcription translation. The gene will undergo transcription translation. Some enzyme is made, but at the end, if you see, the enzyme is non-functional. It's not able to transform the substrate into product. We call this a non-functional enzyme. Transcription translation happened, but the enzyme which is formed is non-functional. That's the second possibility. And the third possibility is, in such a manner mutation happened that the modified allele was not able to produce protein itself. The modified allele was not able to produce enzyme itself, means no enzyme synthesis. So what is the fate of the modified allele? Theoretically, the modified allele can make any one among these. The first one, it can make a normal enzyme or little less efficient enzyme. The second one is going to make a non-functional enzyme. The third one is going to make a no enzyme at all. Now if you go with the first case, the modified allele is also making the enzyme, normal enzyme. That means enzyme is formed, enzyme will transform substrate into product, and such type of combinations are common in nature. Whereas go with the second possibility and the third possibility, non-functional enzyme, no enzyme. When there is non-functional enzyme, when there is no enzyme, then the substrate cannot be transformed into product. That means it remains passive. It's not able to express. Now if it remains passive, the modified allele, when it remains passive, as in the case of second condition and third condition, then the transformation of substrate to product depends upon this unmodified allele only. So because this remained passive, it's producing non-functional enzyme or no enzyme, then the transformation of substrate depends on the unmodified allele only. That is why the unmodified allele, which is expressed in every condition, is called as dominant allele, and the modified allele, which is not able to make an enzyme, which is not able to cause transformation of the substrate, is called as a recessive allele. So in this manner, NCERT, the author tried to explain us what is dominance. They asked exactly what is dominant. Why are that some alleles dominant and why are some alleles recessive? First you should understand what is gene they are telling. Gene is a one which carries information to express a particular character or a trait. In diploid organisms, two copies of genes will be there. That means two alleles will be there. Now these two alleles need not be always homozygous. It can be heterozygous also. Means one capital and one small will be there. Now when one capital and one small is there, now out of this, the smaller one is modified. Capital one is unmodified. The smaller form is modified because of some mutation. Because it got modified, now it changes the information. Now for better understanding, we'll take a gene which is coding for an enzyme. Now it has a pair. one which is unmodified one will make an enzyme. That enzyme will transform the substrate into product and the phenotypic expression is changed. Whereas the modified allele, the modified allele, what is the fate of the modified allele? It can make three. It can make normal enzyme or less efficient enzyme. It can make non-functional enzyme, or it can make no enzyme. Now, if in the first category, when it makes a normal enzyme, then that normal enzyme transforms the substrate, and such combinations are common in nature. Whereas go for the second one and third one, non-functional enzyme and no enzyme, in such case this allele is not able to express. In such case, now it depends on the unmodified allele only. For the transformation substrate, it depends on unmodified alleles. Such unmodified alleles are called dominant alleles, and modified alleles are called recessive alleles. Take a screenshot, then we will talk about codominance. Right. In incomplete dominance, we told the gene is partially dominant. It is not completely dominant, and as a result of that, in F1 generation, the offspring did not resemble both the parents. It did not resemble red color nor it resembled white color. It made a new color which is pink color. Whereas coming to the next concept called as codominance. In codominance, the F1 progeny will resemble both the parents. And the example for codominance is, the best example is ABO blood grouping. So let's talk about the next topic, which is called as codominance. What happens in codominance? Both the alleles can express independently. Both the alleles can express independently, and since both the alleles are expressing independently, the F1 progeny will resemble both the parents. The F1 progeny resembles both the parents. Right, so the example for codominance is ABO blood grouping. ABO blood grouping was discovered by a scientist called Karl Landsteiner. ABO blood grouping was discovered by a scientist called Karl Landsteiner, right? Now, Karl Landsteiner ABO blood grouping, how does it goes? So now, in humans have chromosome number 9. In humans, on chromosome number 9, there is a gene which codes for blood grouping, and that gene is called as I gene. Now this gene, blood grouping gene, I gene, which is present on the 9th chromosome, normally a gene has 2 alleles, right? But this blood grouping gene has 3 alleles. What are the 3 alleles of the I gene? One allele is IA, the second allele is IB, the third allele is small I. So I gene, blood grouping gene, has 3 alleles, IA, IB and small I. Now here we can say that small I is a recessive allele. We know whenever we write small, it means it is recessive allele. Whereas IA and IB are dominant alleles. IA and IB are dominant alleles. So even though there are 3 alleles, even though there's a possibility of 3 alleles, 9th chromosome will be a pair only. Since all of us are diploid, 9th chromosome will be a pair only. So even though there are 3 alleles, there is possibility of taking only 2 alleles among them. So it can be IA, IB, like this combination can come, or anything like IB small I combination can come. So even though there are 3 alleles in an individual, only 2 alleles can come. But if we take a population, in population all 3 alleles are possible. So how many alleles are there? There are 3 alleles. There are 3 alleles. Now with these 3 alleles, how many combinations we can make, how many phenotypes we can make, how many genotypes we can make means we will discuss. Have a screenshot. Right. So with 3 alleles, we can make 4 combinations, 4 phenotypes. And genotypes. Right? So now if the genotype is IA, so if IA genes are there, then the blood group is called as A blood group. If the genotype is IB, then the blood group is called B blood group. If the alleles are IA, IB, then the blood group is AB blood group. And if the genotype is small I, both are recessive, then the blood group is called O blood group. It's called O blood group. Now, it can be this way also. One dominant and one recessive allele also, but since it is dominant, the blood group will be called A blood group only. The other possibility is it can be IB with small I also. Since IB is dominant, then the blood group will be B blood group. So with 3 alleles, what are they? IA, IB, small I. So how many combinations of genotypes you can make means 1, 2, 3, 4, 5, 6. So alleles are 3. Genotypes are 6 genotypes. And the phenotypes are, how many blood groups are there? A, first blood group, B, second blood group, AB, third blood group, O, fourth blood group, right? Again A came, again B came. So that means there are only 4 possibilities. So 4 phenotypes, 4 blood groups. 4 phenotypes, 6 genotypes and 3 alleles. This is a combination. This is a combination. Now we will work out and we will see the Punnett square, parental generations. So have a screenshot of this, then we will get into the Punnett square. Right. So when we have to see the possibilities, how can we check it? With these 3 alleles. Now, parents, since they are pure, so if we take a parent of A blood group, it should be IA. And another parent, if we take B blood group, it will be IB. Since parents are pure, we are taking IA, IB. Now this will make a gamete IA and this will make a gamete IB. Now crossing will result F1. Now in F1 generation, the blood group will be IA, IB. Since IA, IB both are there, so the blood group will be AB blood grouping. That's what I told. In codominance, both A and B are dominant and they express together. And the F1 generation will resemble this parent also, will resemble this parent also. They're having A blood group also, they're having B blood group also. So if they don't resemble either of the parent, incomplete dominance. If they resemble both the parents, codominance. Now, if we do selfing for F1 generation, if we do selfing, then we get IA, IB. So the Punnett square we have to draw this way. IA, IB. IB, IB. Means since IA is there, this is A blood group. A and B are there, AB blood group. A and B are there, AB blood group. Only B is there, B blood group. Now what is this? This is F2 generation. Now what is the phenotypic ratio in F2 generation? What is the phenotypic ratio in F2 generation? 1 A blood group we got, 2 AB blood groups we got, and 1 B blood group we got. So means the phenotypic ratio for F2 for codominance is 1:2:1. And what is the genotypic ratio then? What is the genotypic ratio then? The genotypic ratio is 1 AA. Yeah, it is AA. Means we can write in this manner also. 1 IA, 2 IB, and 1 IB. So if you check carefully, the phenotypic and genotypic ratios are the same. It is 1:2:1, 1:2:1. The same ratio we got for incomplete dominance also. Now this is a Punnett square, and the probabilities when you take parents as pure A and B, then the F1 hybrids are A and B, that's why it's called codominance. And from F2 generation when we get 1:2:1 ratio. Now 3 alleles, since in the normal conditions only 2 alleles will code for a character, but if 3 alleles are coding for a character, that concept is called as multiple allelism. The concept is called multiple allelism. So when more than 2 alleles are coding for a character, then that is called multiple allelism, right? So then what is the formula? How many alleles are there? Basing on that, how many phenotypes and how many genotypes? The formula is N is the number of alleles. Number of alleles. The formula is N into N power 1 by 2. N into N power 1 by 2 is a formula to calculate the number of genotypes. So here in multiple allelism, in codominance, in blood grouping, there are 3 alleles. Means N is 3. That means 3 plus 1, 4 by 2. 3 4s are 12. 12 by 2. So it will be 6. That means 6 genotypes. Yeah, we got 6 genotypes. So if they give you these many number of alleles are there, calculate the number of phenotypes, means calculate the number of genotypes. This is the formula. N into N power 1 by 2, and N is the number of alleles. Take a screenshot, we will discuss. Now blood grouping can be involved in forensic science. Now on blood grouping, they can ask you questions like father's blood group is so and so, mother's blood group is so and so, what are the predictable blood groups of the children, like that they can ask you. So let us see some questions on this concept. Now if the father's blood group is A and mother's blood group is B, what is the possibility of the child blood group? Now you can go like father's blood group like this, mother's blood group like this. If father's blood group and mother's blood group are homozygous, homozygous A and homozygous B, then the child blood group will be, now if you see this with this, this with this, this with this, this with this, so it will always be IA, IB. Means if the parent is homozygous A and other parent is homozygous B, all the children will be AB only. But if the father is like this and if the mother is also like this, then what is the possibility? So IA small I, IB small I. When you make a Punnett square, it will be IA, IB, IB small I, IA small I, small I, small I. Means here AB blood group will come, here B blood group will come, here A blood group will come, here O blood group will come. Means if both the parents are heterozygous, then all 4 possibilities A, B, AB, O came. If both the parents are homozygous, then all the children will be AB only. They'll be AB only. And if the father is homozygous and if the mother is homozygous, let us see this also. If the father is homozygous and the mother is heterozygous, the father will give IA, the mother will give IB small I, then the possibility for the children is IA, IB, IA, IB, IA small I, IA small I. Means AB blood group, AB blood group, A blood group, A blood group. Now if the father is homozygous and mother is heterozygous, then all the children will be either AB blood group or A blood group. So like this they will ask you questions. And if I take another condition, father is A blood group and the mother is I blood group. Father is A blood group and the mother is, or father is, we'll take father is AB father is AB, mother is O blood group. Father has AB blood group, mother has O blood group. Now, which blood group cannot come in children? They will ask you. Which blood group cannot come in children? If the father is AB, mother is O. Let us try and see. So, father IA, IB, mother small I, small I. When you make a Punnett square, it will be IA, I, IB, small I, IA, I, IB, small I. So, if this is the condition, here the blood group is A, here the blood group is B, here the blood group is A, here the blood group is B. Means the children will have A or B blood group, but the children will not have AB blood group, or they will not have O blood group also. They can ask you questions like this. You have to make the possibilities and you have to give the answer. If I give this question that father is AB, mother is O, which blood groups will not come in children, means AB will not come in children, O will not come in children. So like this, on blood grouping, they can ask you questions. Hope you understood what is codominance. Codominance means both the alleles are dominant and they express, and F1 generation resembles both the parents. In codominance, what is a phenotypic ratio and what is a genotypic ratio? It's 1:2:1 only. Example for codominance is ABO blood grouping. ABO blood grouping is also examples of multiple allelism. Why? Usually 2 alleles only will code for a character, but here 3 alleles have coded for a character. So ABO blood grouping is example for multiple allelism also. It's example for codominance also, right? And ABO blood grouping is called agglutination. So in agglutination, right, where are the blood group antigens present and where are the antibodies present? So the antigens are antigen A and antigen B. Antibodies are anti-A and anti-B, right? So blood grouping is determined by 2 antigens and 2 antibodies. Now, where are blood grouping antigens present? Blood grouping antigens. Blood grouping antigens are called as isoagglutinins. Antigens are called blood grouping antigens are called isoagglutinogens, and the blood grouping antibodies are called isoagglutinins. Antigens are always present on the surface of the RBC. Antigen A, antigen B. Antigens are present on the surface of the antibody, whereas blood group antibodies, which are called isoagglutinins, are present in the serum, or they are present in the liquid portion called plasma. Blood group antigens are present in the serum, blood group antibodies are present in the plasma. Take a screenshot. Right. So what is that gene? The gene is called I gene. Where is the gene present? On chromosome number 9. So that gene has how many alleles? 3 alleles. IA, IB, small I. So the allele IA will code for an enzyme. Called glycosyl transferase. Will code for an enzyme glycosyl transferase. So this glycosyl transferase will make a sugar, will make a modified sugar. This modified sugar is nothing but antigen A, which goes and binds to the surface of RBC. Similarly, IB allele will also code for an enzyme. IB also will code for an enzyme. It codes for a different glycosyl transferase. Which will simply add galactose sugar. Galactose means it is antigen B, and this antigen B goes and binds to the surface of the RBC. Whereas the third allele is small I. Small I, since it is recessive, it will not code for enzyme. Since it is not coding for enzyme, no antigen is made. Okay, so now with this possibility, if you see how many blood groups will come, means 4 blood groups we told. What are the 4 blood groups? A, B, AB and O. So in A, which antigen is present? Which antibody is present? In A blood group persons, on the surface of the RBC, antigen A will be there, and in their plasma or serum, opposite antibody, anti-B will be there. In B blood group persons, since IB is there, antigen B will be there, antibody A will be there. In AB blood group persons, both the antigens, antigen A, antigen B, since both the antigens are there, the complementary antibodies should not be there. So AB blood group doesn't have antibodies. O blood group doesn't have antigens. Antigens are not there. Since both the antigens are not there on the surface of the RBC, both the antibodies will be there. Anti-A, anti-B antibodies will be there. So since O blood group is not having antigens on their RBC surface, O blood group is called as universal donor. O blood group is called as universal donor. Why they are called universal donor? Since their RBC is not having any antigens. So if they are universal donors, who are universal recipients, who are universal acceptors, AB blood group are called as universal acceptors. Means O can donate blood to A, B, AB and O also. That's why they are called universal donors. AB are universal recipients, universal acceptors. Means AB can take blood from A also, B also, AB also, O also. They are universal recipients. Take a screenshot, then we'll talk about the next concept called inheritance of two genes, dihybrid cross. Occasionally, a single gene codes for more than one product. Then what is the scenario? Occasionally, when one gene produces more than one effect. So what is an example? With which example we can understand this in a better manner? So take the pea grain. So if we see the start synthesis in the pea seeds, the start synthesis in the pea seeds is controlled by one gene, and that one gene has two alleles. The two alleles are capital B and small b. Now the two alleles can be homozygous for capital B, they can be homozygous for small b also, they can be heterozygous for one capital and one small b also. Now, if it is homozygous for capitals, capital is dominant and small b is recessive, then since capital B is there, the start synthesis is more effective. It is effective. As a result, they produce large starch grains. Since starch is more, the size of the grain will be bigger, and at maturity, the shape of the seed is round. See, one gene is showing two changes. What are the two changes? The first one is the start synthesis it's controlling the same gene. The shape of the seed also it is controlling. So this is called as pleiotropy. When one gene shows more than one influence, when one gene shows more than one phenotypic characters, that's called pleiotropy. So here when capital B is there, since both capitals are there, the start synthesis is effective and bigger starch grains are formed, and at maturity the shape will be round. Let us see what happens for small b. Since both are recessive, the starch synthesis will be less, so the grain will be small, and at maturity the shape is wrinkled. Okay, now come to the heterozygous condition and see, means one capital and one small. So in heterozygous condition, one capital and one small. Since one capital one small is there, so it will not be too much effective or it will not be too less. The starch synthesis is at moderate level, is at the middle level. So that's why the size will be intermediate size. So that means if you take the size is not big, the size is not small. That means in this parameter, so here this is not resembling this parent, not this parent. That means it is incompletely dominant. In terms of starch synthesis, it goes incomplete dominant because it's not big nor small. It is incomplete dominant. Whereas in terms of size, now at maturity the seeds are round, means the shape, it is obeying this shape only, means in terms of shape it is dominant. In terms of shape, it is dominant. In terms of start synthesis, it is incompletely dominant because it's not matching this size also, not that size also. Now, basing on this, we can come to a conclusion. Therefore, we can tell that dominance is not an autonomous feature of a gene also, of the product also. If I'm writing any capital letter, so don't always think that it is a dominant gene. So, we should tell that dominance is not an autonomous feature for the gene also, for the product also. Then on what it does it depends? It depends as much as on the gene product also, and it depends upon the production of the particular phenotype also. Effective production are, less production are, intermediate production are. It depends as much as on the gene product also, and it depends upon the production rate also of the particular phenotype. It also depends upon the particular phenotype that we choose to examine. If we choose capital T and small t, then this concept is not there. Dominant and recessive is only there. When you choose capital B and small b, then the scenario is changing. So we are concluding and we are telling that dominance is not an autonomous feature of a gene or the product. It depends upon the gene product. It also depends upon the production rate of the particular phenotype, and it depends upon the particular phenotype that we are choosing to examine. When these three sentences are applicable, in the case when one gene is influencing more than one phenotype. If one gene is influencing more than one phenotype, what is it called? The concept is called pleiotropy. If one gene is controlling more than one phenotype, one gene is controlling the start synthesis rate also, it is controlling the shape also, it is called as pleiotropy. So I think you understood this concept. When one gene is controlling more than one phenotype, it's called pleiotropy. The plant example for pleiotropy is P synthesis, start synthesis. The animal examples for pleiotropy, we can take sickle cell anemia, we can take phenylketonuria. We'll study that in genetic disorders chapter. Take a screenshot of this, then we will talk about dihybrid cross. The next concept is dihybrid cross. It can also be called as inheritance of two genes. Means Mendel conducted cross between the pea plants for two characters. So Mendel has taken, so the parents are taken as, so I'll erase this, dihybrid cross. We need a bigger board. So remember that we are talking about dihybrid cross. So Mendel has taken parents as one parent is round and yellow, another parent is wrinkled green. So round means capital R, yellow, capital Y, capital Y. Wrinkled means small r, small y, small y. Now, so when we do crossing between them, from here the gamete will be capital R, capital Y. From here the gamete will be small r, small y. And when we do crossing, the F1 generation will be capital R, capital Y, small y. So the F1 are hybrid. They are double hybrids. They are heterozygous for both the characters. Since R is there, they are round. And since capital Y is there, they are yellow. Means the F1 plants resemble round and yellow only. They resemble round and yellow only. Whereas the other ones, if you take, they are wrinkled and green. They are wrinkled and green. Now when F1 plants are selfed, now for such a combination, how many types of gametes are formed? There is a formula which is called as 2 power n, and n is number of heterozygotes. N is number of heterozygote condition. So here R is also heterozygous, Y is also heterozygous, means 2. That means 2 power 2 means 4 types of gametes we get. What are the 4 types of gametes? So we can write it here. The 4 types of gametes are capital R with capital Y, capital R with small y, small r with capital Y, small r with small y. Similarly, this side also will be the same, capital R with capital Y, capital R with small y, small r with capital Y, small r with small y. So when we draw the Punnett square, 4 into 4, 16 combinations we are going to get here. Now, the first one is capital R, capital R, capital Y, capital Y. Capital R, capital R, capital Y, capital Y. The second one is capital R, capital R, capital Y, small y. Capital R, capital R, capital Y, small y. The third one is capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Continue for the remaining. Capital R, small r, capital Y, small y. Here you have to combine this with this. Means capital R, capital R, capital Y, small y. Capital R, small y, small y. Capital R, small r, capital Y, small y. Capital R, small r, small y, small y. This with this means capital R, small r, capital Y, capital Y. Capital R, small r, capital Y, small y. Small r, capital Y, capital Y. Small r, capital Y, small y. This with this means capital R, small r, capital Y, small y. Capital R, small y, small y. Small r, capital Y, small y. Small r, small y, small y. So these are the phenotypes. Now when we have to write the genotypes, capital R means round. Capital Y means yellow. That means this is round, yellow. RR is there, means again round. Capital Y is there, yellow. R is there, capital R, then it will be round only. Capital Y is there, then it will be yellow only. R is there, round only. Capital Y is there, yellow only. Here also capital R is there, round only. Capital Y is there, yellow only. Here also capital R is there, round we get, and capital Y, yellow. Next one also, since capital R is there, round we get, and capital Y, yellow we get. And if you see here again capital R, capital Y means round and yellow we get. Again here if you see capital R, capital Y means round and yellow we get. Now coming to the other one, only capitals are there for R, Y is small, means capital R for round, small y if you go it is green, round and green. Here also capital R means round, Y means green. This line also capital R means round, small y means green. Now then small r is coming. If small r comes, then it is wrinkled. And capital Y means yellow, wrinkled yellow here. Small r again wrinkled. Capital Y yellow. Small r wrinkled. Capital Y yellow. See the last one. Last one all four smalls like this means it is wrinkled and green. It is wrinkled and green. So this is a 16 combination of F2 generation. Now if we have to show the phenotypic ratio, if we have to show the phenotypic ratio of dihybrid cross, so then count how many round yellow are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 we are getting round and yellow. How many are you getting round and green? 1, 2, 3. So 3 we are getting round and green. How many are you getting wrinkled and yellow? 1, 2, 3. So 3 are wrinkled yellow. Last one we got wrinkled green. So if anyone asks you what is the phenotypic ratio for dihybrid cross, means 9:3:3:1. 9:3:3:1. In this 16, 9 plus 3, 12, 12 plus 3, 15, 15 plus 1, 16. In this 16, how many parental are there? Means how many are round yellow or wrinkled green? So 9 are round yellow, 1 is wrinkled green. Means 9 plus 1 means 10 are parental. The remaining 6 are recombinants. 6 are recombinants. Means what do you mean by recombinant? If round is going with green recombinant, if wrinkled is going with yellow recombinant. So this is a phenotypic ratio. We can calculate the phenotypic ratio in other manner also. Take a screenshot, we will see that also. Now the same phenotypic ratio we can get in other manner also. Dihybrid cross means, is it two monohybrid crosses? Now means monohybrid cross for one character, that is color, monohybrid cross for other character, that is shape. So that means you will get 3 round, for shape you will get 3 round, 1 wrinkled. 3:1 into, if you take for color, 3 yellow is to 1 green. So you are going to multiply 3:1 into 3:1. 3 are round, 1 is wrinkled, 3 are yellow, 1 is green. So if you multiply 2 monohybrid cross also, you will get 1 dihybrid cross. So 3 into 3, 9. 3 into 1, 3. 1 into 3, 3. 1 into 1, 1. So this 9 are 3 into 3, round and yellow. Round and yellow. So 3 are round and green. 1 into 3, wrinkled and yellow. 1 into 1, wrinkled and green. So this is a phenotypic ratio. It is 9:2, 3:2, 3:1. If they ask you the genotypic ratio, you have to split the 9 as 4:2, 2:2, 2:1. Split the 3 as 2:1. Split the 3 as 2:1. Split the 1:1. How many types of phenotypes you got? 4 types. 9, 3, 3, 1. How many phenotypes you got? 4 phenotypes. How many genotypes? Come on, count. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 genotypes, 4 phenotypes we got. The phenotypic ratio is 9:3, 3:1. The genotypic ratio is 5:2, 2:2, 2:1, 2:1, 2:1. So take a screenshot. Then we will talk about the law which supports dihybrid cross. After proposing this dihybrid cross, Mendel proposed the second set of generalizations, which is called law of independent assortment, which supports the dihybrid cross. We will see that. Take a screenshot. So that law states that when two pairs of characters are combined in a hybrid, one pair segregates independent of the other pair. After working with dihybrid cross, Mendel proposed the second set of generalizations, which is called as law of independent assortment, which supports the dihybrid cross. The law of independent assortment states that when two pairs of traits are combined in a hybrid, when two pairs of traits are combined in a hybrid, one pair segregates independent of the other pair. One pair segregates independent of the other. Means if you take a hybrid, capital R, small r, capital Y, small y, this is hybrid. So when two pairs of traits are there in a hybrid, one pair segregates independent of the other pair. Means if it is a double hybrid, how many types of gametes will come? 4 types of gametes will come. 2 power 2, 4 types of gametes will come. In this 4 types, now 2 times capital R will go, 2 times small r will go, 2 times capital Y will go, 2 times small y will go. So this is how you get 4 types of gametes, because one pair will segregate independent of the other pair. So 50% if capital R is coming, 50% small r will come, 50% capital Y is coming, 50% small y will come. Because of this proportion only, so if you count, you will get the ratio as 9:2, 3:2, 3:1. So law of independent assortment supports dihybrid cross. Law of dominance and law of segregation supports monohybrid cross. Take a screenshot, then we will get into the next topic, that's chromosomal theory of inheritance, proposed by Sutton and Boveri. If we have to discuss dihybrid cross, what will be the ratio? We'll just see now. So the next topic is dihybrid test cross. So recollect, what do you mean by test cross? If we know the phenotype and if we have to determine the genotype, we have to take that and we need to cross it with a recessive parent. So in dihybrid cross, it is capital R, small r, capital Y, small y. It can be this way, or it can be capital R or capital Y. So we need to find out whether it is this genotype or that genotype, because round and yellow phenotypes we know. It is round and yellow. If it is heterozygote also, it is round only. If it is heterozygote also, it is yellow only. Or when it is homozygous also, it is round only and yellow only. So we do dihybrid test cross to find out whether it is homozygous or heterozygous. For that we need to cross it with a recessive parent, small r, small y. Here also we need to cross it with a recessive parent, small r, small y. So we know that this is going to produce only one type of gamete, that is small r and small y. Here we get four types of gamete. Capital R will go with capital Y, capital R will go with small y, small r will go with capital Y, and small r will go with small y. So when we make the combinations, this with this, this with this, this with this, and this with this, we get the ratio as capital R, capital Y, small y, this combination. Capital R, small y is the second combination. Small r, capital Y, small y is the third combination. This with this will be small r, small y. So when we have to write the phenotypes, this will be round and yellow, and this is round and green, and this is wrinkled yellow and wrinkled green. So we got all four types in 1:1:1:1 ratio. So if you get 1:1:1 ratio, then we tell it is heterozygous for both the things. And let us see here, what do we get? So here we get only one type of gamete, that is capital R and capital Y, since it is homozygous. Here we get small r and small y. So when we fuse them, we get capital R, capital Y, small y. So all are round and yellow only. So if all are round and yellow, so what is your inference? The inference is the unknown genotype is homozygous. And if you get round yellow, round green, wrinkled yellow, wrinkled green is 1:1:1:1 ratio, means the inference is it is the unknown genotype is heterozygous. The unknown genotype is heterozygous. Here if all are round and yellow, the inference is the unknown genotype is, isn't it homozygous? Yes, it is. It is homozygous. Now, what is test cross? Test cross means if you know the phenotypes, we need to check whether it is homozygous or heterozygous. If it is homozygous, it will be capital R, capital Y, capital Y. If it is heterozygous, it will be capital R, capital Y, small y. So we need to check the genotypic combination. For that, we do test cross. Then in test cross, what do? We take that organism whose genotype is to be determined and we'll cross it with a recessive parent. And when you're crossing with a recessive parent, and if you get the combination of all four in 1:1:1:1 ratio, the inference is it heterozygote. If you get all round in yellow, your inference is it is homozygous. So take a screenshot, then we'll talk about the next topic called chromosomal theory of inheritance. Yeah, in NCERT, under chromosomal theory of inheritance, so they started telling why Mendel's works were not recognized. With that introduction, they tried to explain us the topic of chromosomal theory of inheritance. So we will also start the same way as we are following NCERT line to line, so that it will be very much scoring and helpful for your NEET examinations. So let's go to the topic chromosomal theory of inheritance. chromosomal theory of inheritance, right? So the author tried selling that Mendel published his works in which year? Mendel has published his works in Normal Journal, Society of Brunn Journal in the year 1965. He published his works in a journal called 1965. But for several reasons, Mendel works remain unrecognized till 1900. Mendel published his works in the year 1865, but they remained unrecognized till when? They remain unrecognized till 1900s. Now, if you ask what are the reasons for Mendel's works to not to be recognized means, so in NCERT textbook itself they gave 3 reasons. Firstly, they will tell communication was not easy those days, means journals and all or the print media and all is not too much aggressive at the time. So the first reason for Mendel's work to not to be distributed is communication was not easy that days, and his works are being published in a local journal only, so it did not go on to all the loop and corners of the world, right? So that's the first reason they told. And the second reason, Mendel is telling that the factors which we now call them as genes are stable, discrete, they don't undergo any changes. But at the time the contemporaries are too much influenced by Charles Darwin's variations, right? When they're able to see variations, so they were not able to understand this, how he's telling that they are stable, they're discrete and they're just passed on to the generations. That is the second drawback. And the third drawback is Mendel, since he has a mathematical background, he started applying mathematics to solve the biological principles, which was unacceptable for many of the biologists. And finally, when they asked Mendel to show the physical proof where genes are located, so he couldn't give the physical proof. Because of that also Mendel's works are unrecognized only. Why he was not able to give physical proof means at the time microscopes are not there, right? If microscopes are not there, then how can you see a cell, how can you see a nucleus, how can you see a chromosome, and then on top of the chromosome gene, how can you show it? So we cannot tell that Mendel couldn't show it. At the time that technology is not there to see the chromosome and to see the gene. Finally, because of all these 3 or 4 points, we can tell that Mendel's works remain unrecognized till 1900. Then we can tell that in 1900, 3 scientists worked on the same works what Mendel worked, and 3 people from 3 different countries independently worked and they proposed the same principles what Mendel laid. Now what are the 3 scientists' names? The first scientist is Hugo de Vries from Holland, and the other scientist is Correns from Germany, and the third scientist is von Tesmack from Austria. Now these 3 scientists worked on the same principles what Mendel worked, and they gave the same conclusion what Mendel also gave. So that means if one person is proposing, okay, he might be wrong. If the second person is also telling the same thing what Mendel told, and the third person is also telling the same thing what Mendel told, then they understood that like Mendel is correct. We were not able to understand at that time, but yeah, what Mendel proposed is correct. And then they converted the Mendel's rules into Mendelian genetics, or they converted them into laws of genetics, right? And by the time in 1900s, microscope is also designed, right? So when microscope is also designed, with advancements in microscope, we were able to study cell division. By the time, due to advancements in microscope, due to advance in microscope, they were able to see nucleus under microscope. They were able to see nucleus under microscope. And they were able to see nucleus, and they're telling that at the time of cell division or just prior to cell division, means in the I phase, in the S phase, the nucleus will double and in the cell division the nucleus will divide. And when they are looking into the nucleus, they saw that there are some colored bodies. Those colored bodies are called chromosomes. Those colored bodies are called chromosomes. These chromosomes double in the S phase, means the DNA content doubles in the S phase, and in the cell division they segregate or they separate and they get distributed in the daughter cells. So because of the advancement of microscope, they were able to observe the nucleus, they were able to observe the chromosomes under microscope. So then in 1902, two scientists, their names are Sutton and Boveri. So in 1902, two scientists, Sutton and Boveri, they noticed that the behavior of chromosomes is parallel to the behavior of the genes. Why not? Because these are the chromosomes, and on the chromosomes, if capital Y, small y genes are there. Now, if the chromosomes are moving, then the gene which is present on the chromosome will also move. If I am sitting in a bus and if the bus is moving, that means literally I'm also moving, right? It's the same concept. Sutton and Boveri in the year 1902, they observed that the behavior of the chromosomes is parallel to the behavior of the genes, and they used this chromosomal movement to explain the Mendel's laws. These people, they observed that the behavior of the chromosomes, movement of the chromosomes is parallel to the behavior of the genes, and they used this chromosomal movement only to explain the Mendel's laws, right? So they tell that chromosomes or genes are present in pairs. We are diploid organisms. Now in diploid organisms, the chromosomes are in pairs only, right? This is one pair of chromosome, and if we have to see the other chromosome, this is another pair of chromosome. So they tell that the chromosomes are present in pairs, and genes are also present in pairs. Now genes has alternating forms of genes are called alleles. We call them as alleles. Alleles are present on pairs, and the alleles for that gene are present on the same loci on the homologous chromosome. For example, if you take the gene which is coding for the height of the plant, T gene. So T gene is present at the same loci, capital T and small t. If you have to take about the seed shape, capital R and small r. So they are present on the same loci on the homologous chromosomes. The genes are present on the same loci on homologous chromosomes. They are present on the same loci on homologous chromosomes, right? And take a screenshot, we will continue. Now these chromosomes during metaphase, they align on the equatorial plate, and in anaphase they move towards the poles, means we can tell they segregate independent of each other, they are telling. So we need to understand how the chromosomes move, in which stage do they move, we need to understand, then we can tell if chromosomes are moving parallelly, genes also will move. So in NCERT, let me take 4 colors. In NCERT, they have explained 2 possibilities. Possibility 1 and possibility 2, and they say that the chromosomes are present in pairs. They are arranged on the equatorial plate in meiosis 1, in metaphase 1. In NCERT, by mistake they have given anaphase, but then aligning on the equatorial plate, it will be in the metaphase stage only. So they are trying to explain with 4 different colors. In possibility 1, they have taken taken 4 colors, orange color and green color. For orange color, I'll use this. And green color they have taken, I have green. And parallelly on other side they have taken yellow color and red color. For yellow color I'm going to use blue. And for red color, we are going to use red only. So this is a pair of homologous chromosomes, and this is a pair of homologous chromosomes, right? So then on the metaphase plate, they are aligned in this manner. With the help of spindle fibers, they are aligned on the metaphase plate in a proper manner. Now, this is the arrangement of chromosomes. Suppose if we have to understand the gene movement also, we will take that here if capital Y is present, then on this homologous chromosome small y will be there. And here if capital R is present on the green chromosome, then it's on the other homologous chromosome small r will be there. So this is during metaphase 1 of metaphase 1 of meiosis 1. It's metaphase 1 of meiosis 1. Then what happens by meiosis 2 in anaphase? By meiosis 2 in anaphase, meiosis 1 anaphase we need to write. So at anaphase they divide. So when they are dividing, they divide into 2 cells. So this is one cell and this is the other cell. In one cell, in NCERT, this is given which color? Orange color. And this is green. And here this is orange color, green color, yellow color it is with red. They tell that orange is segregating with green and yellow is segregating with red. So that means we need to show orange color is going with green color and yellow color, means I'm using blue, is going with red color. Right? So that means capital Y is segregating with capital R here, small y is segregating with small r. So it's in meiosis 1 anaphase 1. And if we have to see meiosis 2 of anaphase 2, meiosis 2 of anaphase 2, this is anaphase 1. So again this cell will split into 2 cells. Parallelly this cell also will split into 2 cells. This cell is splitting into these 2 cells and this cell is splitting into these 2 cells. Now at the centromere the chromosome will split and it distributes like one here and the other here. This also splits at the centromere and they divide like this. One green chromosome, daughter chromosome, the other daughter chromosome. Here what happens? The blue colored chromosome splits one chromosome and another chromosome. The red chromosome also splits red and red. Means it is going capital Y with capital R, this also capital Y with capital R, this is small y small r, this is small y with small r. So here they are telling that the orange colored chromosome is segregating with green and the yellow colored chromosome is segregating with red in one possibility. And we can check the other possibility also where they will go orange with red. We'll see that possibility also. So all this is to tell that the behavior of chromosome is parallel to the behavior of gene. Okay. So here for orange color, since I don't have orange color marker, I'm using black color. In the possibility 2, orange color is with red color. It's with red color. And then the yellow color, since I don't have yellow color, I'm using blue color. The yellow color will go with green color. And these are the spindle fibers. So on the black one, what chromosome we have? So we are having capital Y. So capital Y is now going with small r. Here capital Y is there with capital R. Here capital Y is there with small r. And here small y is there with capital R. Now this is in meiosis 1 metaphase 1. So this is at metaphase 1 of meiosis 1. Now what happens in anaphase? In anaphase they move, right? Meiosis 1, anaphase 1. So after meiosis 1, after anaphase 1, they split and they make 2 cells. At the end of meiosis they make 2 cells. Now one chromosome, this chromosome comes this side and it segregates with the red chromosome. the black is segregating with the red chromosome here, and blue is segregating with the green chromosome. Blue is segregating with the green chromosome, means small y is going with capital R, capital Y is going with small r. Now if we have to see after meiosis 2, we'll get 4 cells, 2 cells from this and 2 more cells from this. Now here the centromere will split and this chromosome will divide into 2 daughter chromosomes. Red also will divide into 2 daughter chromosomes. The blue chromosome also will divide into 2 daughter chromosomes, and the green chromosome also will divide into 2 daughter chromosomes. And capital Y is going with small r, capital Y going with small r, small y going with capital R, and small y going with capital R. So if you see that the chromosomes, when they are moving, the genes or the alleles which are located on them, they are also parallelly moving. So this is a diagrammatic explanation of the chromosomal theory of inheritance. Chromosomal theory of inheritance was proposed by Sutton and Boveri in the year 1902. So they observed that the chromosomal movement, during meiosis. This is meiosis. During meiosis, the behavior of chromosomes is parallel to the behavior of the genes, and they tried to explain the Mendel's laws with the help of chromosomal movement only. And these are the 2 possibilities they have given to explain. Take a screenshot, then there's a table. We'll try to understand that table also. Right. So in the table, they try to give us how are they same, in which points this chromosome behavior and the gene behavior is different. And then they asked to identify which 3 points are for chromosome and which 3 points are for gene. So let us see that we are writing gene here and we are writing chromosome here. So the first point what we can tell is, for board exams, if they ask you explain chromosomal theory of inheritance, so if you write that chromosomal theory of inheritance was proposed by Sutton and Boveri, and it explains that the behavior of genes is parallel to the behavior of chromosomes, and if you write these 3 points, you'll get full out of full. So what is the first point it says? They tell that the genes occur in pairs. Yes, in diploid organisms, genes occur in pairs and chromosomes also occur in pairs. That's the first point. The first point is genes occur in pairs and chromosomes also then occur in pairs. Genes occur in pair and chromosomes occur in pairs in diploid organisms. Now just now we saw in the previous picture that they are segregating. When are they segregating? Diploid organism becomes haploid organism during meiosis at the time of gamete formation. So when they are segregating at the time of gamete formation, only one of a pair is transmitted to the gamete. Among capital R, small r, only one will go to the gamete. Among capital Y, small y, only one will go to the gamete. So we can write they segregate at the time of gamete formation, means genes, they segregate at the time of gamete formation, means during meiosis they segregate such that one of each pair is transmitted to the gamete. One of each pair is transmitted to the gamete. So genes are segregating at the time of gamete formation by meiotic division, so that among capital R, small r, only one will go to the gamete. Among capital Y, small y, only one will go to the gamete. In the same manner, where are the genes present? On the chromosomes. That means chromosomes also segregate at the time of cell division only. Same point is written there also, because it is the same, right? That's what they're trying to explain. The behavior of chromosomes is parallel to the behavior of the genes, right? So the same point we can write here. Chromosomes also segregate at the time of gamete formation only. Chromosomes also segregate at the time of gamete formation such that one of each pair, out of pair of chromosomes, only one chromosome will go to the gamete. One of each pair is transmitted to the gamete. Now let us try to understand one simple thing. So when we take a pair of chromosomes this way, and when we take another pair of chromosomes right, so here if you see capital T and small t are there, and here if capital R small r are there, and at this end if capital Y small y are there. So T and R are close to each other, they are linked, that means they are dependent pairs. T and R are dependent pairs, whereas T and Y are independent pairs. So dependent pairs and independent pairs will not occur in chromosomes. Dependent pairs and independent pairs word occurs in gene only. See here, capital T and capital R are very close to each other on the same chromosome. So wherever capital T is going, since they are linked genes, capital R also will go. That means they are dependent pairs. Whereas capital T is at that extreme and capital Y is at this extreme, means they are independent pairs. So here we tell that the independent pair segregates independent of each other. Dependent pairs will not segregate, but independent pairs segregate independent of each other. That's the third point. The third point is independent pairs, means this and this. Independent pairs segregate independent of each other. So whereas for chromosomes, what we can write, in chromosomes there are no independent and dependent pairs. One pair of chromosome, other pair of chromosomes, black chromosomes and blue chromosomes. One pair segregates independent of the other pair. For chromosomes we can simply write the third point as one pair segregates independent of the other pair. That's it. So if they ask you what is chromosomal theory of inheritance, what is the significance, who proposed it, how can you explain, means with the help of these 3 points you can explain. And in NCERT they asked, after giving these 3 points, they asked find out which is gene and which is chromosome. I started writing this as gene and that at chromosome only. The first 2 points are same for gene and chromosome. Genes are in pairs, yes they are in pairs. Chromosomes are also in pairs in diploid organism. This is a pair of chromosome, this is a pair of chromosome. At the time of gamete formation, since gametes are haploid, reduction division, meiotic division should happen, and one chromosome only will go to the gamete. That we wrote there. They segregate at the time of gamete formation and one of each pair is transmitted to the gamete. Here also the genes are also present at the time of gamete formation. They segregate and only one will go. When coming to the third point, I tried to show you what are dependent pairs and what are independent pair. T and R are independent, T and this is T and R are dependent, T and Y are independent. Independent pairs segregate independent of each other, means it is gene. One pair segregates independent of the other pair, means that is chromosome. So take a screenshot, then we'll continue with the same topic, chromosomal theory of inheritance. After observing the chromosomal movement during meiosis, Sutton and Boveri argued that the pairing and separation of the chromosomes will lead to pairing and separation of genes also. That's what they argued. Sutton and Boveri argued that the pairing and separation of chromosomes would lead to segregation. Segregation means separation only. Segregation or separation of the genes also. Those genes which are present on the chromosomes will segregate along with the chromosomes only. And finally, Sutton is the person who united the knowledge of this chromosome chromosomal separation, chromosomal segregation with Mendelian principles, and he called it as chromosomal theory of inheritance. If they ask you who united the knowledge, means it is Sutton. Who proposed it means Sutton and Bovary together. In which year means 1902, right? And then who gave the experimental verification for chromosomal theory of inheritance? It is Thomas Morgan Hunt. Thomas Morgan Hunt has given experimental verification for this theory called chromosomal theory of inheritance. Who is Thomas Morgan Hunt? He is an American geneticist. He got Nobel Prize for discovering linkage and recombination in the year 1933, and he produced many stalwart students like Student, he produced Bridge, he produced Muller. Student, Bridge, Miller are the students of Morgan only, and he's often considered as father of experimental genetics because he has given the experimental proof for chromosomal theory of inheritance. He is popularly called as father of experimental genetics, and he worked on an experimental material called fruit fly. He is also called as fruit man or fly man of genetics. He is the person who saw that if two genes are nearby on the same chromosome, then they are called linked genes. So he is the person who coined the term linkage, and he is the person who coined the term recombination also. So as I told you that Morgan's experimental material is fruit fly. The scientific name of fruit fly is Drosophila melanogaster. Now what are the advantages which are present in the fruit fly which helped Morgan to work out the experimental things? Means we can see that fruit fly can be easily grown on a simple synthetic medium. In lab you can easily grow it in a simple synthetic medium, or if you bring any fruit, a ripened fruit, thousands and thousands of fruit flies will come into the home, right? So it can be easily grown in a simple synthetic medium. So it is cheap to cultivate it, right? And the second point is it completes its life cycle in 2 weeks. Means within 2 weeks you will get the results of the experiment what you have conducted. It completes its life cycle in 2 weeks, and single mating produces many progeny, many offsprings it will produce. And the third one, clear distinction of sexes. In NCERT they have given the diagram of male Drosophila and female Drosophila. Male Drosophila is smaller, female Drosophila is bigger. So means sexual dimorphism, clear cut differences are seen between male and female Drosophila. And then many hereditary variations, many contrasting characters are seen under low power microscope itself. Body color, eye color, wing size. So there are so many contrasting traits which can be easily observed under low power microscope. And then these are the 4 points of fruit fly they have given NCERT. And if we have to tell few more points, we can tell they are very small. So it means they are not occupying space. They are 2 millimeters in size, and they possess only 4 pairs of chromosomes. Among 3 are autosomes, and the 4th one is allosome. The allosomes are like us only, XX, XY, sex determination. XX for female, XY for male. The allosomes in male Drosophila is X and Y. The allosomes of female is X and X. So this is the remaining part of chromosomal theory of inheritance. Take a screenshot, then we will talk about linkage and recombination, the concept explained by Morgan. Right. Morgan conducted dihybrid cross experiments in fruit fly. So he conducted dihybrid cross experiments in fruit fly. He has taken the X chromosome. Which chromosome did he take? He has taken the genes which are located on the X chromosome. So he conducted dihybrid cross experiments in fruit fly. So he has taken the genes which are present on the X chromosome of the Drosophila. So the first thing, he has taken the body color. He has taken the body color and eye color. So if he has taken this as a male, he crossed it with which body color? Yellow body, white eye is crossed with brown body and red eyed female. So he has done cross for 2 characters which are present. Both these genes are present on the X chromosome only. So when he has done cross for 2 characters, means it is dihybrid cross. What 2 characters? He has taken the body color and the eye color. So body color, yellow body is a recessive one, brown body is a dominant one, white eye is a recessive one, red eye is a dominant one. So when he crossed and when he's checking the F2 ratio, he was expecting dihybrid ratio as 9:3:3:1, but the ratio what he got is completely deviating from this ratio 9:3:3:1. He got a different ratio. So what are the ratios he got? Means let us see. So when he has taken yellow body with a white eye, it is cross 1. So I'll erase this. So we'll take the full board. We need the full board here. He has done 2 type of crosses, cross 1 and cross 2. So he selected male and female Drosophila. Since, as I told you, he has selected the genes which are present on X chromosome, female will have 2 X chromosomes and the male will have, we'll use a different color. Female will have 2 X chromosomes and the male will have 1 X and 1 Y chromosome. So he has taken the 2 genes which are present on the X chromosome. First he has taken yellow body, white eye. Yellow body, white eye, female it is. Yellow body, white eye, female. So this is a mutant variety. Mutant variety means it is a recessive variety. And the male, what he has taken, so he has taken a dominant one. Dominant one means it is brown body. Brown body means Y plus. Red eye means W plus. So he has taken the genes which are present only on the X chromosome. That's why on the Y chromosome we are not going to represent anything. He saw the genes which are present only on the X chromosome. So Y plus denotes brown body and W plus denotes red eye. And this is a dominant one. This is a dominant variety, or we can tell it's a wild one. Wild variety. So Mendel crossed these 2. These are parental and he'll get F1 generation. In F1 generation, he'll get 1 female and he'll get 1 male. He'll get 1 female and he'll get 1 male. So male will pass on the Y chromosome to the son. So this Y chromosome will come to the son as it is. And the son will get X chromosome from the mother. It means the son is getting this chromosome from the mother. Means Y and W. Now the father will pass on the X chromosome to the daughter. Means father is passing on this chromosome to the daughter and the mother also will pass 1 X chromosome to the daughter. Means this X chromosome came to the daughter. So this is Y and W, and that one from the father what it came is Y plus and W plus. Since this is dominant, here the daughters are brown body, red eye. And since he got this gene from the mother and this Y chromosome doesn't have anything, so here the sons will be Y for yellow body and W for white eye. So his F1 generation is like this. When the F1s are intercrossed, so when the parents are crossed, he got F1. When the F1s are intercrossed, we'll get F2. Now in F2 they have given 4 males and 4 females. 4 males in 1 square, 4 females in 1 square. So we'll draw all 4 males first and then we'll go for 4 females. All the 4 males will get Y chromosome from the father. Means Y chromosome, Y chromosome, Y chromosome, Y chromosome from the father. And the mother will give the X chromosome to the sons. All these 4 are sons. So mother will give the chromosomes, means mother will give the same green chromosome, right? Means Y and W, Y and W. Now this is parental, because Y and W is parental, Y and W is also parental. Sometimes what happens during recombinations, Y will go with W plus and Y plus will go with W. So that is recombination, right? So in recombination, how should we represent W? And this much part I need to use Y plus, and here I will use, yeah, so here I'll use W plus is coming with Y. So if you see, out of these 4 males in NCERT, they have given this under one block. Out of these 4 males, these 2 males, Y with W is parental, whereas Y plus with W, W plus with Y is recombinant. Let us see for daughters also. We'll draw one more block and we will see for daughters. And we know for daughters, the father will pass on the green chromosome, right? So that means all daughters will get one green chromosome sure from the father. And that will be Y and W, Y and W, Y and W, Y and W. So mother will pass on one chromosome to the daughter. So the mother will pass on Y and W. Mother will pass on Y plus W plus, right? So here also it will be Y plus and W plus. So mother will pass on this chromosome to one son and this chromosome to one son, Y plus and W plus. The same we have done. Green chromosome came here, blue chromosome came here. So this is parental, this is also parental, this is also parental, this is also parental. What is recombination? Recombination means if this is blue and next one green comes on, means Y plus is coming with W, and the other one Y is coming with W plus. So 1 plus and 1 normal, 1 plus and 1 normal are recombinants. So if you see carefully, so these 4 are males since Y chromosome is there. These 4 are female progeny since both are XX chromosomes only. Out of this, this one, this one, this one, this one, all these 4 are parental, whereas plus with normal, normal with plus, plus with normal, normal with plus are recombinants. Mendel saw when he has conducted such a type of cross, now these 4 are recombinants, these 4 are parental, the recombination percentage he got 1.3. Recombinants, he got how much percentage? 1.3. That means 100 minus 1.3, 98.7 are parental only. How much is the parental combination? The parental combination he got 98.7. So why did he get that much huge amount of parental combination? Means he saw that Y and W, yellow body and white eye genes are very close to each other. They are very close to each other on the same chromosome. Since they are very close, the linkage, the strength between the 2 genes is more. If the strength is more, if they are nearby, if the strength is more, then they don't participate in crossing over recombination. So then the recombination percentage will go down only. It will be 1.3 only. So to check that, he has conducted another cross. This time he has taken another cross. This time he has taken females and males. He has taken the characters which are present, the genes which are present far away. So he has taken W and M. Okay, so W is eye color and M is the size of the wing. So what is W? Come on, W is white eye. And M is miniature wing, small wing. And this is recessive. Since plus is not there, it is recessive variant. Or you can tell this is mutant. And he crossed this with a male. So for male, Y chromosome, and we have to take the X chromosome. The X chromosome is having W plus M plus. So W plus will be red eye, and M plus will be normal wing. So this is dominant or wild type. This is dominant or wild type. So when he has done cross between the two genes which are present on the same chromosome, but see the difference, they are present far away from each other. One is at this end, other one is at that end. Means they are far away from each other. That means the strength between them is less because they are far away from each other. So if you do a cross like that, what happens? Let us see. In F1 generation, one daughter and one son. For son, father is going to give the Y chromosome, right? And mother is going to contribute X chromosome. So means this chromosome will come, W and M. For daughter, mother will give one X chromosome. And father means W and M. Father will give one more X chromosome. So the father has given this chromosome Y plus and M plus, right? So she is daughter. Now here the daughters are red-eyed, normal wing. M plus came, so normal wing. And here the sons are white-eyed, W is there, so white-eyed, and miniature wing. Now when he has intercrossed F1 generation, same he got F2 generation. He got 4 males and 4 females, like that he got 4 males and 4 females. In this cross also he got 4 males first, then 4 females. Now all the males will carry the Y chromosome from the father. These 4 are males, I am telling. That means they will carry this Y chromosome from the father. So Mendel did not work out on the genes present on the Y chromosome, so that's why we are simply drawing Y chromosome like that. And these 4 boys will get X chromosome from the mother. So the mother will give red chromosome for one son, and the mother means W and M. The mother will give another chromosome Y plus and M plus to another son. W plus and M plus to another son. And during meiosis, during gamete formation, recombinations will happen. Then what happens? With M plus means with W plus M can come, or with M plus W can come. So W with M is parental, or W plus with M plus is parental, whereas 1 plus and 1 normal, 1 plus and 1 normal is recombinant. Let's talk about the girls also, daughters. Now in daughters, father will give this X chromosome for all the 4 daughters. Means I need to draw as it is. Father has given 1 X chromosome to all 4 daughters with W, M, W, M, W, M, W, M. So mother is going to give this X chromosome to one daughter, same W and M. Mother is going to give the other X chromosome to the other daughter. Means W plus M plus. Now recombinations happen the other case. In recombinations, W will come with M plus, and so we can write here, sorry, red color we need to use, right? So red we are using, yeah, M plus and W, and here W plus and M. So if W is going with M, if W plus is going with M plus, so these are parental. If W goes with M plus or W plus goes with M, these are recombinants. When he has done cross 2 for 2 different genes which are present on the same chromosome but they are far from each other, then he got the recombination percentage. These 4 are recombinants. Why are they recombinants? Normal is coming with plus, plus is coming with normal, normal is coming with plus, plus is coming with normal. So these are recombinants. He saw the recombinant percentage as 38.2. He saw the recombination percentage as 37.2. 37.2 minus 100. If you do, you will get the parental combinations. 1, 2, 3, 4 are parental combinations. So when you do 37.2 minus 100, the parental combination, it will be 62.8. It will be 62.8. So when you do dihybrid cross for the genes which are present on the same chromosome, which are near to each other, then the strength is more, then the recombination percentage will be 1.3 only. And when you do a dihybrid cross for the two genes which are present on the same chromosome but they are far from each other, then the recombination frequency will increase from 1.3 to 37.2. Parental combinations will decrease from 98.7 to 62.8. So he called this type of genes which are parental genes as linked genes. He called these genes as linked genes, and those combinations which are non-parental, Y plus with W, as recombinants. So these are recombinants and these are linked genes, and he told the strength of the linkage will determine the recombination. If they are tightly linked, recombination will be less. If they are farly linked, recombination will be more. So this is about recombination. The next topic is sex determination. Take a screenshot.